Can you
Start.
Hi everyone, thanks again for joining us today on Research Vessel Falcor for another ROV Sebastian dive on the East Pacific Rise. And today we are back in Teddy Bear site, which is one of the vents that we've been exploring with a series of scientific experiments. And I will hand you over to one of our lead scientists to explain more about what we've been doing. Stefan?
Yeah, thank you. Good morning. This is Stefan Sievert. I'm a scientist from Schmidt Oceanographic Institution on Cape Cod in Massachusetts in the US. And I'm a marine microbiologist, and I'm on this cruise to look at microbes, their diversity and function. And I'm part of the team that also explores the subseafloor biosphere, and we are interested to see if the subseafloor can also be a habitat for other organisms besides microbes, including Riftia larvae, for example, that then actually come from the subseafloor and colonize these vents. So for the last week or so, we have been setting out experiments to test exactly that hypothesis. And today we're going to start out a little further north at the site called Teddy Bear to pick up an experiment that I actually left back here on a cruise in spring of 2021. But then we will make our way to south and check out on the experiments today and see what it looks like, and also do some other sampling along the way. So yeah, happy you can join us today, and I think we have an exciting day with a lot of different things to do and things to explore and see.
So, is this what you want to start looking at, or?
Yeah, so I think the site that we look for actually is a little up here.
Right. See it, just imagine kind of rocks on your left. Yeah.
Yeah, there you see the marker
Yeah, I see it.
back here. Yeah, perfect. So we can maybe do a quick survey of that area. So this is a site that we actually discovered on a cruise in 2014. And at that time, this site was characterized by warm venting from the subseafloor, and there were no animals, no mussels, no riftia, and the rocks were colonized by filamentous bacteria, so it had this appearance of a fuzzy rocks, and so that's why some of us in the science team named it actually Teddy Bear. But since then we have been visiting the site frequently, and there have been colonization by riftia and also mussels, but right now it looks like that the site actually is less active than it was a few years ago. But you can see here an experiment that I left here. We're going to just check out what it looks like. So what I was studying at that time was the degradation of the tubes of the tube worm of Riftia, and see what bacteria are involved in this and how it's broken down. And so I left here a mesh with inside pieces of the tube worm. And that tube is actually composed of chitin and also some proteins. And it's very hardy and hard to break down, but some bacteria obviously can do it. So we'll see. That looks like actually there's not too much left of it. Yeah, but the plan is now to pick this up and to put it either in one of the sampling boxes that we have. Can we zoom out a little bit? Oh, yeah.
Yeah.
Okay. So the question is, I think the idea was to put this in one of the bio boxes, but one possibility could also be in a coral river, but I don't think it would fit in there, right?
Not without folding it up.
Yeah, but I think the question is also with the marker, right? That might sort of be difficult to put in.
Yeah.
So might we have to go with the bio box? I think it might be the easiest
then we'll
way.
put it anywhere you like.
Yeah.
I know when I talked to Sabine, One of the thoughts was later on they were going to potentially keep the front bio box for if we had a smoker, so we could tip it into there for collection. I don't know if that's still the case, but
Yeah.
we might want to leave that front one available if they want that.
Yeah.
I'm not sure.
Yeah, I mean, I think for the chimney piece, we might want to do it and put it in the coral quiver. I mean, we could try and see. I mean, if the rope would stick out of the coral quiver, it might not be too bad. If we can actually squeeze everything in and just the rope would stick out, that might not be too bad. We could try that, maybe.
We could, but if the rope is sticking out, we can't put a cap on there.
Yeah.
It'll block the cap.
Yeah.
Also, that's attached to it, right?
Yes, that's the weight
Do
actually
you know what
that
the
keeps it
diameter
down.
that is?
Maybe like this.
Ooh, that
It might be all. Yeah, let's put it in the bio box. I think that's easiest.
Yeah.
Don't spend too much time. So we'll put it in the bio box number 3. But it really looks like that the tube pieces that I had in the mesh are not really there anymore.
So we want to put it in bio box 4, right? Because 3 we potentially want to leave open for a smoker.
So there's another small. Oh.
Yeah.
Oh yeah, yeah. Okay, we put it in bio box 4.
Okay,
Okay, I
just
changed
making
it. Yeah, no, I think that's a good point.
Have that.
So you got that, Andre?
Yeah, so sample 70.
I'm not sure. It's not a sample. It's actually picking up an experiment.
I can bring it back in so you can
This would be a sample, no? Or not?
That's a good question.
Yeah, I think it is because
Okay.
you do studies with this, no?
Yeah.
Or analyze.
Yeah.
Yeah. Let's go box 4.
Mm-hmm.
Yeah.
No, just roll it. I think you got it out.
Great. Thank you. And before we leave, maybe we can like just do an overview of the site,
Sure.
like a flyover, and maybe collect some video.
Yeah.
Yeah, it really looks like that this site has become less active. So there was a time when we discovered it, and then it was more active colonization of riftia and mussels eventually. But right now it seems like there's not much activity there anymore.
There's one rift here. 2.
But there's still some of these rocks that actually have this fuzzy appearance by filamentous bacteria that are attached to them. So these bacteria are sulfur oxidizing bacteria, so they actually live at the interface, so they are attached to the rocks and then get the sulfide from the fluid that comes up and at the same time get the oxygen. So their metabolism from the seawater. So their metabolism is very similar to the symbionts of Riftia that some of you might have heard about during this cruise already. So Riftia houses symbionts inside in a special organ, the trophosome. And these bacteria are also sulfur-oxidizing bacteria. In that case, they actually live inside the worm and get everything delivered to them. And here, these are free living and have to find it themselves.
With that overview.
Yes, that's great. Thank you very much. And so now we would move south, and there's a site that we discovered yesterday. That we call, I think it's a waypoint. I forget the waypoint, but it was called Fuzzy Rock.
South?
Yeah, south on the way to Tika.
Okay.
Yeah, this one exactly. Yeah, great.
So, can you grab the vessel, move it somewhere about right there? Maybe it's not there. You can also just get a range and bearing.
Yeah,
Longer.
like 30 meters out.
Yeah, well, let's do more than that.
70 meters. Bridge, Roby.
Go ahead, RV.
Can we get a move 70 meters due south? We're trying to figure out our vessel position on the map.
Yeah, 70 meters down. RV, that's in progress now.
After that, thank you.
So, what do you think this
Yeah,
is?
so I think, I just wanted to talk about that actually. So I think what you see here, this orange colored sediment, I think that's mostly also caused by bacteria that actually oxidize iron. So these are iron oxidizing bacteria, and then they basically oxidize reduced iron to form iron oxides, and that is basically the color of rust. So it's different than the sulfur oxidizers that actually are white due to the sulfur that they produce. And yeah.
Very cool.
There must be some venting, very small venting here potentially, that causes the appearance of these iron oxidizing bacteria.
Great job.
Any idea why we wouldn't see that in other venting sites? Just that one?
Yeah, yeah. I mean, I think it's the chemistry differs sometimes, and so when we see the other vent sites, they're heavily dominated by sulfur,
Yeah, I've
sulfide,
got that.
and so that's why you have bacteria that actually grow on the sulfide, and that's the white that sort of predominates in that case.
Okay, cool. So heading down to the, can you wait?
Yes, please.
Can see the depth.
This guy is just chilling.
Yeah. Anyone know what that is? It's like a worm? Or
That's a sea cucumber.
sea cucumber. Yeah.
See-through.
Yeah.
If you poke it, it will puke out the stomach.
You're asking us to poke it?
Sir?
You asking us to poke it?
No,
Just you know, but normally, I don't know, deep sea cucumbers, but normally they regurgitate all their inner organs when you poke them. It's a defense mechanism.
Okay.
Yeah, everything that is inside they do.
So this was a giant hole we were flying in.
Yeah. It's at the bottom of the trough, and so there we're gonna measure the temperature, but don't take any fluids. Just measure the temperature of the flow coming out, and then we're gonna take a couple of slurps or suction samples.
Okay. Look at like the height, like the 60-ish. See if there's so much rain going on here. It is there. It's the other half for it. The other half of the S.
Yeah, there's this big sea anemone that we have also seen on our way sometimes. So I think we're getting close.
That's good.
I'll just come back out a little bit.
Oh, okay.
I got it.
That's it back there. There's more up on the top too, I think.
Yeah, I think it was further down in the.
That's the first stuff that we saw
Okay,
there.
yeah.
And we inspected that crack, but I think there was a larger section up above. You can see it up here.
Yeah,
But we can just kind of do an overall. That way you can choose your best site. What do you like,
yeah.
if you want?
I believe there was something like further down in the trough. No, I think we can maybe fly over a little bit.
Sure, we can try it, but I think that was it right there.
It looked a little different yesterday when I was.
So let's move back up. That's flat now. That's going up on the other side. There's that big hole we inspected. We moved on to it. So yeah, that was for sure it.
Yeah, what's down there? This here. I think that's it.
There's
On the other side.
I don't think we can
Over.
even fit in there.
Oh,
So
we're just at the area with the fuzzy rocks. Do you remember? Is this the place? Or
No, there was a lot better than that. So,
It was this.
oh, in
In position now.
like over here?
Is this close to the
Hmm?
or do you want a little bit further
right
south?
on the edge.
It was
On the edge?
face.
Okay, so you okay?
It was this right here.
Yeah,
It's that okay?
that's more like it.
All right.
And a little higher up.
So yeah,
Racers.
this
Okay.
is what caught our attention, remember? But there was
Yep.
that crack right up
Yeah,
here.
that's
We
All
inspected
right.
that.
Okay.
And then there's also more up above,
But this is where we saw
right there.
things.
Okay, so we
and
should
more things
yeah
there.
go there.
Probably,
Okay, yeah. Okay.
Have a quick look.
Thank you, Stefan.
You're welcome.
Thank you. You were right.
No problem.
So the worms, I mean, all the stuff I've seen,
Yeah.
they were there
Position now.
in this
Right.
darkened area. I'll come with my computer in about
Okay.
easy to reach, maybe.
Thank you.
So it looks like there's two hot spots there and there. Which
Yeah.
one do you like better, the left or the right side?
Yeah, I mean maybe we can sort of measure temperature like in both places,
Okay.
and then I'm not sure. Is it possible to do it?
It's big enough that we might have to move out.
Okay.
I'll see if I can set up where we do it.
Okay. That would be great.
We might.
I think this side looks maybe a little bit better for slurping and maybe easier access to it. So I think maybe go for this side first. So what we see here is hydrothermal fluid coming out of a crack in the seafloor. We don't know the temperature yet, but definitely there's some sulfide associated with it. So we have these filamentous bacteria that live at the interface. They take up the sulfide from the fluid that comes up and then take the oxygen from the seawater. So I would expect this is like maybe slightly above ambient temperature. We'll see. And then at the same time you have the bacteria, and there are of course other organisms that will eat on it. So we have here some limpets and then also scale worms that will then again eat on the limpets. So it's like a food web based on bacteria. Chemosynthetic bacteria, as they are called, because they can, they oxidize sulfide with the reduction of oxygen, and that generates the energy that they need to fix CO2, similar as plants use light to fix CO2. These organisms use the energy from so-called redox reaction to fix CO2. And they're at the base of the food chain down here at the vents. So we are now setting up for measuring temperature. Okay, yeah. So just temperature, no fluid at all.
Is this a null site?
Yeah, no, we just named this yesterday, so it's waypoint 17, fuzzy rock. And so we're now setting up to measure temperature. Yeah, I think we're going to just focus on this side probably. Yeah. So try to find a place where we have a lot of shimmering water. It's always an indication that fluid with a different temperature and different density is coming out and mixing with the seawater. And you can also see that different types of bacteria. So we have these filamentous bacteria that give the appearance of fuzzy rock, but then we also have these sort of more like globular shaped aggregations that are different types of bacteria. So it's just not one type, but there are a variety of different self-oxidizing bacteria. So now we're measuring the temperature.
Up here. So we're showing 3.6. They're working on it, but I can show you in the meantime while he's getting it up. You can see it right here. It's 1.9, 2.5 right here.
Okay, so it's slightly, okay, only slightly above. Maybe go a little bit into the, yeah, okay, yeah,
7.8,
nice, okay,
4.
nice, okay, yeah, stay there.
Cool.
Oh, that's actually pretty warm. Wow, I'm surprised. So currently we're measuring around 9 degrees centigrade, which is significantly above the ambient deep sea temperature of like just 2 degrees. So there's definitely, you know, fluid coming out. Okay. Maybe move a little bit in here, in this area.
You can see there. On the top there. 80 to 10. 8.5. Yeah.
Like over here. Yeah. Okay, so I think it's, yeah, that's, so we measure around 9 degrees, up to 9 degrees, close.
That's the hottest we got right there.
Yeah, so it's warm, yeah,
8.94 is the
yeah,
hottest.
great. So I think, yeah, we can stop the temperature measurement.
Okay.
And so next we would continue with the suction sampler, and we would put it on mesh. Let me see. Very fine mesh, number 4.
Pause 1 second. Is
Mhm.
there at any point that we're going to use the mass spec bag that we're using?
No,
for
not
this
here.
area?
No,
Not here?
not here. Yeah. Thank you for asking.
Sure.
So with this next suction sampler, so we have a very fine mesh. I think that, I believe that's like 34 microns, which is of course larger than bacteria might be, but these are filamentous bacteria and they clog up. So hopefully we'll retain some of these bacteria on the mesh. So we're going to try to slurp or suck up some of these filamentous bacteria.
Are you wanting to use the ones that have your mesh?
Yeah, the very fine mesh, number 4.
Was that 4
Yeah, we have it on number 4 and 5, I think.
or 5?
Yeah, so we can start with 4. That would be sample 71, right?
The samples are this is site as well.
Be careful going right because that's pulling that hose down and doing that push that causes problem. Don't go right.
Such a set of 3.
4.
4. This is the fine mesh.
Very fine mesh, yeah, very fine.
That one. I think you just push down until you see it.
Of course, that's always interesting because, like down here, when we measure like something like 10 degrees or 9 degrees centigrade, we call it a warm spring. But that's all relative because the ambient deep sea temperature is 2 degrees. So relative to that, it's warm. But of course, on land, we will probably still call this a cold spring, not necessarily a warm spring.
Yeah, I mean, I guess in this position it would be yaw because you're so far over. Yaw would bring you down, not pitch at this point. Though. I didn't know that. the side grabs in between will give you more dexterity. A little bit less reach, so it's kind of lead. Go for a drop. Yeah, you're finding that.
So we are now in the process of getting the suction sampler ready, and then we'll try to dislodge some of these bacteria from the rocks and slurp them up. Some video.
Other way. It's working. If you go the other way and rotated, it would just come straight down on the site. It might be a little easier.
Maybe you can sort of go here and can you zoom in a little bit? Oh, I can do that. Yeah, thank you.
Yeah, I'm just trying to give the pilot what he needs to see.
Mm-hmm.
Sometimes he's got to see his arm to know what
Of course, yes, yeah. That always has priority.
So you're wanting to vacuum in that darker area there? Right.
Yeah, I think we can sort of start over here, these filaments here, and then we go in here, yeah.
Alright, let's go in.
I might have to scratch the rock a little bit, to get the bacteria off. They're probably quite well attached, so you have to sort of scrape, yeah. Yeah. So as we can see, these bacteria actually are very well attached to the rock where they live. And they can withstand the force of the suction sampler so far.
It's not much. But
Yeah, I know. Okay, yeah, no, there's some material coming in. Yeah, we can also move along a little bit and then pick up some of this material over here. Maybe it's also possible to pick up a piece of the rock at the end. Suction seems to be a little difficult to get the bacteria. Yeah, but maybe move a little bit over here.
Remember with that fine mesh, that
Yeah.
slows the flow of the pump.
Yes, of course.
So
Yes.
maybe a little bit
Yeah.
I don't know if that makes a difference.
I think maybe if it's possible to go to this here.
So I think it's
All right.
possible yesterday when we flew
Oh yeah,
here-
there's some material now coming off. That's good.
Yeah.
Yeah, great. Nice.
I was gonna say, I think it's possible when we flew here yesterday that
Yeah.
when I backed up, some of that light stuff blew away. Some of that stuff we're getting now.
Yeah.
So the spot up above, we never did go look at. So
Yeah.
there might be some good collections there.
Okay. Yeah, I think we focus here, and then because we have characterized now the temperature, and then yeah.
Sure. Okay.
But that's yeah. Thanks for pointing that
No
out.
problem. Oh, there you go.
Yeah. So we were able to dislodge some of that, but I think the filaments are really tightly attached to the rock, so it's difficult to get them. So maybe in the end we'll try to break off a piece of rock and put it in one of the sampling devices that we have. Yeah, so I think we can stop that now, probably.
Okay.
And maybe just,
Wait till that little bit
yeah,
gets in there.
exactly.
Okay.
To chamber number 6, and then get some of these, like here, like limpets,
Okay.
Some of
we are in
these.
6.
and some scale worms to Stefan here.
Yes.
Okay, so maybe you can point. I think there's some limpets here, right? That are,
Yeah, and you see the scale worms on the face that is orange as well. And there's one under the overhang close to the limpets.
yeah. I mean, if you want to come here and
Sure.
just point it out.
This is which success?
That's still
Which success sample we're using now? Is that
number 6.
number 6.
These are not the next for you.
Oh no.
Sorry, bit chill. So these are the scale worms we're aiming for. And limpets to the left, and there's another scale worm hanging
Oh yeah.
in there
Okay, great.
as
Yeah.
well. And the red one, you have a little more to the right after that.
Okay, turn on the suction. Good luck, those things usually hold on pretty.
Yes, they do. And it's slightly
That's
overhanging,
number
which makes it
2,
just
right?
a little more
Yeah.
challenging.
Yeah.
This is 72. Success sample 6.
Maybe easier ones a little slightly to the right. I saw some more earlier.
Success sample.
We'll come back to that one. Oh, it's moving now. Oh, bastard.
Run!
It's like, oh yeah, you're gone.
All right.
Yeah. Yeah.
It's hanging there.
Yeah.
There's more to the right, so
Okay.
it's hiding now. There's one, and there's another one there. And
Oh, this one here. Yeah.
yeah, and
and
another
then
one further right. Yep.
yeah. Okay.
The ones I've seen yesterday.
This is a large mesh, no?
Yes.
It is
Course.
large.
Course. Oh,
I got that one.
There it goes.
Yeah, excellent.
And go for this
Good
one.
legs.
And there's another one here.
Get them.
did we already? Then we can maybe get some of the limpets, right?
Oh yeah, and we need the limpets.
Didn't see him make it into the jar.
You didn't?
No.
It's.
I think.
So
let's-
It probably
Yeah.
is under the overhang. Try maybe the one
I'm
on the
watching the jars. He sometimes they'll hang on to a ledge. and
into the
I'll keep them flow on until we get them. But
Plus, they're actually not as big as they look.
Mm.
You guys have a pretty good zoom on that thing.
yeah, true.
You think you got it, Alex? I was not even paying attention. Yeah.
One.
Okay, cool.
And then we can try
Yeah,
to
the
get these
limpets there,
limpets.
and there's a scale worm just under the overhang, right behind your section sampler there. But that's, I think, in there for good for now. Let's go limpets first.
Be a little tough. Yeah, from past experience, we have to scrape those super
Yeah.
slow.
Right, you guys know where to get these. Oh, I saw something flying. Looks like a scale worm.
Like scale worm.
And there's another one out there. You can see it
Okay,
crawling around.
different
as well.
one. Has some bacteria on it.
I'll stop over and go.
Work. They look fine.
Yeah,
It's
scale.
around, but it can.
Or that too. You have that tool. You ever need it? For stuff like this. It really helps.
Sorry, say that again.
You want to know? Yeah.
Yeah, there's another scale worm over here. Seems like a little tricky to get the limpets off, right?
Yeah, they're not easy.
Yeah. It's here, right here.
Yeah, guys. We have pros at this now.
What do you think? Is that enough?
And don't
Should we
think
try?
the limpets got it in there.
Yeah, I know, right?
He's
It's-
almost, with
but
the
it seems-
scale worms, almost got it. Almost.
Oh, that's it's down here.
Still here. Where is it? It is still here.
But it seems like it's hard to get the limpets off.
Yeah, they're definitely a little challenging.
Oh yeah.
Yeah.
Hopefully got in there.
Might have some. Yeah, we got one. I see one floating around in there.
Okay. Maybe one last attempt to add the limpets. Probably have to. It's hard to get them right to actually scrape them off, but it's not easy to access. Did you see any other place where there are limpets that might be more accessible? Maybe we can.
Yesterday we just checked that out real quick and then moved on because we were
Yeah, right.
on a time crunch. But there may be more. There was another gray area to the right of this, like 1.5 meters to the right of this. So maybe.
Yes. I think we're hitting. There's a little bit of a ledge.
Yeah.
Yeah, so I think it's probably not. Yeah.
So I mean, you can
Okay.
try top and coming down, but you're gonna
Yeah.
have less visibility and just super thin. And then as long as this suction has enough power to still pull it in.
Yeah, they're kind of fun. It's like hard to make it out with these little guys. Like,
Too steep a man.
but it
Yeah.
almost looks like they have a little
little flatter, I would say.
but can't
One of them look shiny and bicolor, and that's really cool.
yeah.
Yeah.
Yeah. That little shade.
Yeah, maybe let's stop that here and then maybe move a little bit to the right if we
Yeah,
can
no
do
worries.
that.
There's that
I'm gonna
other
leave
area
the suction
that
on
we
so there's
have.
anything there.
If there's anything, we can take a look. Might be also hard to get in there.
We've got a fishing section.
That'd be cool.
There's something just off to the right too. I think it's a sea cucumber.
No, that's a fish.
No, even further right.
Off of the right, sorry.
This guy right here.
Oh, yeah,
Oh, a fawn.
I saw it.
Yeah, but I don't see any.
On the face of the rock, I see 2 red scale worms there. Closer.
Here?
Probably scale, yeah, they're right there.
Yeah, okay, here, yeah.
They're there,
no
and
limpets,
probably
right?
are,
Yeah, third one there.
yeah, and there's another one. But if you suction maybe at the bottom, you also will get some animals down there.
Yeah. Yeah, maybe just
probably.
suck down here.
Good. Let's also make sure we're not spending too much
Yeah,
time
I
on
know.
it.
Yes.
They're I'm aware.
Sorry, folks, let's go on this first, or?
No, just use the suction sampler down here and then we stop that. It's fine.
There's a rock for it.
Yeah. Yeah, we can maybe.
It's awful big, but.
Would that would not fit in a quiver, right? That would be too big. Yeah.
No.
Put it with,
Oh, big scale worm.
Yeah.
Yeah. Sorry. Big scale worms like that.
Yeah.
It made it
Oh,
to the
Yeah.
to the jar. That's still like swimming around. Swirly.
But remember, we have those milk crates. Those are excellent for rocks like that.
Yeah, but we're interested actually on the bacteria that are on top, so we would need to put them in a closed container.
In the buy box.
Yeah. No. Yeah. No. Not in the bio box. I mean, I think we need a rock that we actually would put in a quiver, in the coral quiver. Okay, let's stop the slurping and then maybe try to pick up like a small piece of rock.
Come on. Sounds like it. Oh, that's my fault. That's good. It's fine. It'll just rest on it a little bit. Yeah, but they want it in a coral coater, so let's get one of those open.
Number 3.
You want it in coral?
Oh, number 1 is. I think number 2 is leaking, as far as I know. Heard. So we can use number 1. Yeah.
Yeah, number 2 is.
So we can use number 1 first. Yeah.
I'm gonna slide up while we're doing this. Slid a little bit. There's that sea cucumber. Oh, get away, get away, get away. Okay. Pull that guy up. Right there.
Yeah, I'm not sure. Like some of these smaller pieces here. How easy? I mean, maybe with a scoop. No, it's not. No, it's not too tight in there, right?
It could be.
I mean, if you can pick up like a small piece.
No, not sure. You can try it.
I mean, if this one would fit, that would be like perfect.
Sure.
Would be an easy grab, right?
You might be a little tight on the rope right now. It's part of my problem. I got it.
So we are now trying to see if that rock that came loose might actually fit in one of the coral quivers that we have, which would be perfect. So we can see it has quite a bit of this filamentous bacteria attached to it. That would be a nice sample. Maybe, yeah, zoom in on it a little bit. Yeah.
You want to, let's get some beauty shots. Over near the camera. On the line. There you go.
Nice.
Autofocus. There we go.
Okay, great. Awesome. Yeah, and that's also, amazing in a way if you think about it, because normally you think of bacteria that you cannot really see, of course, so you need a microscope. But here you see actually bacteria with your naked eye. But of course here the zoom helps as well. But yeah, all of these individual filaments are contained of chains of cells, but it's one filament. Okay, great. Yeah, let's see if it fits. That would be perfect. Might be a little too large.
Yeah.
Eric, get down there with your hammer.
Chisel.
Let's do a breaker.
Come on.
It's gotta be close. If it doesn't fit, it's
really close. If it fits, it ships. Try to grab those kind of easy, or just running around. Yeah, I don't really usually have to like.
Yeah, I think it's too big, right? So
Just kind of like compress it over there.
yeah.
So what if you just get to the right of it and just tap it off to the left? We can't use it anyways, right?
No.
Someone comments that this is a teddy bear rock, and I do think this is the sort of bacteria that's the namesake of teddy bear vent then, yeah?
Yeah, it's similar types of bacteria. You know, we don't know for sure if it's the same. I mean,
Right,
there
yeah.
are different types of filamentous bacteria
Sure.
that are growing under different conditions. Like the temperature plays a role, of course, the sulfite concentration. But in general terms, yes, would be this similar type of bacteria.
Yeah, okay.
So maybe you can leave it open and. Or is that now broken? That okay. Okay, do one last attempt maybe in getting a smaller rock and then put it in quiver number 3. But then we need to move on really. Yeah. Oh, that would still be tight, no? Is that quiver? Number 1?
I mean, I can't guarantee it with that.
Yeah. Yeah, okay.
For anyone just joining us, for today's dive, we are north of the Tika Spire area that we've visited on a lot of our earlier dives this expedition. We're going to continue to head up a bit. We're sort of in an area we visited yesterday, which we were calling the suburbs of Favaflow. But we're going to head up even further up, I believe. We are about 2.5 kilometers deep still, though. And this is on the East Pacific Rise. So an area of the East Pacific Rise. We're about 2,000 kilometers to the west from Costa Rica, and then about 1,000 kilometers south of the coast of Mexico. For folks who have never tuned in for any of these dives, maybe this is your first one. This is live footage. This is actually coming from a remotely operated vehicle named Sebastian. Think of it as sort of like it's a submersible robot. And so it's not a human-occupied vehicle, right? There are not people inside the submersible, but instead this submersible is actually controlled over a very, very long wire. We have a 6 kilometer long wire. Now, only just about 2.5 kilometers of that wire are actually spooled out. But on the other end, right, we have the robot, and then up here is research vessel Falcor 2. And we're in mission control. There are 2 pilots that steer and operate the robot from the surface. And so we send commands down the wire, and the robot sends data and footage back up the wire, and then we can, of course, share that out over the wide web. So if this is your first time joining us, welcome. Type questions in the chat, and we try to answer those as we can. Certainly don't get to all of them, unfortunately, but we do try to get most of them. And we're pretty early still in the dive here. Probably got just shy of like 7 hours left on the bottom would be my guess. Although we've been ending dives a little early if we finish out all of our objectives early enough. So.
Nice.
Fingers are all busted up now, so yeah.
Yeah, that's not really, yeah. Not sure if it's possible to grab something over here. This one.
Let's get some,
Yeah, okay.
We can get a couple
Yeah,
of those, huh?
yeah, sure. Nice. Yeah, maybe one more piece and then we can move on.
Is that the piece there, or?
Yeah, that might not, yeah.
No, I moved around and trying to get you some angles in. So, oh, that's what I'm talking about. That pad, when you did instead of the fingers, yeah, I would pliant, yeah. There you go.
Oh, that's
That's
nice.
super
That's a nice
cool, though.
piece. Yeah, that would be perfect.
Yeah, that was not your fault. That's cool.
Ah,
That stuff is like
yeah.
super brittle.
Now we could slurp.
So there's a little flaky bit right there. Once again, that's going to break up, but you might be able to get a little bit out of it.
Okay, that's one last attempt, and then we have to move on now.
Got it. Oh. Nice. Nice.
Yeah, perfect.
Awesome.
Nice. Nice catch. Maybe, yeah. Great.
So, a question in the chat: if the filaments kind of grow over a known rate, or I assume it would be based on certain nutrients in the water that they'd grow
Yeah.
quicker or slower.
Yeah, I mean, I think, yeah, that's exactly right. I mean, they, as I mentioned earlier, I mean, they really grow at the interface. So they attach the rocks, and then the water, from below, that the fluid that comes up, that contains the sulfide is passing by, and then at the same time it's mixing with the seawater that has the oxygen. And so they really grow at that interface, and they oxidize the sulfide and reduce the oxygen, and that energy from that reaction can be used to fix CO2. So they're autotrophic organisms, as we call them, similar to plants, just they use chemical energy.
Right.
And then they grow, yeah, they can, but then at the same time there are also other organisms that like to eat them,
Sure.
like the limpets, for example.
Yeah.
And so sometimes you can actually see, grazing tracks where they are being grazed, and so yeah, they would grow up to a point, but then they're also being eaten.
Yeah. Yeah. The we saw some of those grazing tracks on the boxes. We
Exactly.
thought,
Yeah, we're going to see that later on again. So we're going to check them out as well to see what they look like.
Yeah.
So, but nice work, by the way. Really nice catch of the rocks. Thank you very much. It's a nice sample.
So 3-phase 440 is the power, and we're sending 4,200 volts down that
number 3, right?
down the cable.
Yeah, okay.
for the questions there about how much power. Quite a lot of power.
So now we are wrapping up our work here at this site that we named Fuzzy Rock yesterday. And then actually talking about the experimental sites, that's where we're actually going to go next to see what they look like today. And as we talked about that yesterday after the dive, and we might actually recover one experiment tomorrow, depending on what it looks like today. But it seems like it's ready to terminate that experiment.
And those the filaments, they're not an elongated bacterium, but they're a chain of bacterium that just keeps
they're
adding on.
individual cells that are attached to each other. So
Okay.
it's, that's also how they grow, basically. Yeah.
Perfect.
But still, it's it's quite cool to actually see them with your naked eye.
It was. Do you know, is that filament like multiple cells wide, or are they like one cell
No, it's
wide?
one cell wide.
Wow,
Yeah,
okay.
Yeah. And maybe, when it comes up, we can actually take photos in the microscope and then maybe, put that also. Online. I think
Yeah, let me know, and maybe we can
I mean,
slap that up tomorrow.
I'm very fascinated by these organisms, and I think they're beautiful and under the microscope. But yeah, microbes can be very, very beautiful.
Yeah.
And I like also the way they kind of wave in the flow and in the mixing zone. It's nice to watch. Yeah, there's some more up here. So now we actually move to lava flow suburbs, to the mesh works. Yeah.
The vessel's gonna get moving.
Right. Okay. Yeah. Of course. Can do a little overview of this area here.
Yeah, we're going back down. Bridge, are we?
Go ahead, Howard.
We've got another updated waypoint.
That's cool how it shifted that up just so sheer like that.
Yeah.
Imagine the force it takes to do this. Or was it? I guess it was molten when it did that. Huh?
Kind of oozing up, yeah. Like, yeah.
Are we going
must have
to
been
the
bizarre.
mesh works, to the experimental site?
How long this will take? Otherwise, they're
You got time for coffee?
Yeah, Crab Spa, for the title of the stream, that's actually a location we're going to. There's an event here named Crab Spa, so we are going to be heading there.
Howard, that's the program.
And as we move along now, you can actually see here the ocean crust, and that's brand new ocean crust that formed after the last volcanic eruption that we had here in 2006. And in some cases, you really see the shiny, glassy surface. That's basically the time. And as the ocean crust moves to both sides away from the rich center, sediment accumulates and it gets thicker and thicker. But what you see here is brand new ocean crust. That's like 16 years old in some cases. One six. If I'm not mistaken, these bags are actually used as weights by the French submersible Nautil. So rather than actually using iron plates, as we have seen before, that are used by the human-occupied vehicle Elven, the French actually use different types of weights.
Huh.
And that's, I believe, what that is actually. So.
That's kind of like interesting to be able to ID the vehicle or the group based off of the weights they're dropping, you know? It's
yeah,
like
yeah, it is. Yeah,
it's looking for sign or spore of like an animal, you know, for tracking. It's like this sub. Yeah, submersible droppings, exactly.
Oh yeah, we have Stefano. That's in here actually, so he could maybe confirm that if that's true or not. But
Yeah, they're now using bags that are made of fabric.
Okay.
That is biodegradable because this can stay forever.
Yeah, for sure. Yeah.
Oh,
very nice
nice squishy ball.
sea anemone here. But some of the iron weight instruments on the seafloor for different purposes, for sampling, for example, and we bring them down with a weight. And at the end of the experiment, when we recover them, the weight is released, and then the instrument has buoyancy and comes back to the seafloor.
Yeah.
And that's also being used in some cases for so-called elevators. So these are sampling platforms that we can use. So they are also being sent to the seafloor, and we can put additional samples on them, and then they shuttle samples or instruments up and down, basically. So it can be used in some cases for more efficient sampling, depending on what is actually being done. But these weights look very similar, or actually the same types of weights that are being used for Elven.
I love flying through these little narrow tracks. Maybe not that narrow. That's a little too narrow.
So now we are on the way to what we have called lava flow suburbs, and so there we have the three experiments that are ongoing right now to test this hypothesis that the subseafloor biosphere does not only contain microbes, but also is an avenue for animals, and especially in the larval form, to go through and recruit the vents. And so that's a very interesting and novel idea that we are testing here. And so we have put up these boxes that are sealed and stained organisms that were already present, but then remove the stain and then look for organisms that might colonize these vents newly and would not be stained. So that's the whole idea. And tomorrow we might actually start removing the first experiment that was put down, I believe, on the second day or very early in the cruise.
Already. Okay. Nice picnic on it there on the screen.
And something that I also have noticed in all the years that I have been coming to the site is that if you're looking for venting, sometimes the crabs can actually be a good indicator. So they are present everywhere, but once you get closer to a vent, they increase in numbers. So that's always a good sign that you are getting closer to a vent.
So I'll put a map up here real quick.
Yeah, here we see some dead riftia.
Read my charts wrong earlier.
This might have been a site that was previously active, and now it has ceased activity.
If you look right above, there's a lot of active.
Yeah.
Just where they're falling down.
So we had started further north of Tika Spire and north of Fava Flow, and now we're headed south down to those. But if I put up this map, you can see just all the different waypoints, all the different markers. We've been all over this site for the cruise so far. Lots of work going on here. And on the camera here, you can see, let me
So we
kick
can
over the
maybe
science.
start. Which one? I think
These
there's
are the
number 1
experiments.
there, maybe. Yeah, so we can look at that first.
From left to right, we have experiment 3, then 2, then 1 here on the far right with the rope bow on top of it.
Yeah, looks like a gift box.
Yeah. Once someone said that, I could not see it now. So
So now, yeah, you see these mesh boxes that I talked about earlier, and we also have here Sabine Göllner, who was leading the efforts in designing these, and so maybe she can also talk a little bit more. But we're checking them out to see what has been growing on the mesh. Like we had these sulfur-oxidizing bacteria just before on the fuzzy rocks, and similar types of bacteria would actually grow here as the interface shifts. So fluid is coming into the mesh box, and oxygen is outside. So they would actually colonize the mesh because that would be a suitable habitat for them, using the sulfide that's coming up, and then the oxygen from the outside. And now you can actually see, some of that biofilm is being stirred up by the sub.
That shows there's a lot of bacteria inside.
Yeah.
all right. So yeah, these 3 experiments have been here for a little while. We visited yesterday and saw some of this bacterial growth. So interesting to see.
Go ahead, RV.
Just shorten up that way by 1.
Yeah, no worries. I'll adjust my hand.
Thank you.
Okay, sorry about that. Camera's all yours, whatever you want to look at.
Okay. Yeah. You wanna maybe? Yeah. Okay.
So Sabine's swapping into camera control.
always so angry.
Stoppst du? Dit is heel mooi.
Das ist erst das schaust.
Mhm.
Okay.
It's number 1. Okay. Can we look at the others and probably afterwards like go to the other side and check on the other. Okay. Did you take a frame grab? Sorry.
Yes, sure.
Okay.
Sure.
So that's leaving experiment 1 and now headed to experiment 2 there on your screen, and not too much further to the left there, experiment 3. So 1, 2, then 3.
That's a scary spot. Not weird because I'm between those. I won't see them. It's kind of a scary spot for that one. I can come
From the
this
other,
way.
yes.
Is that okay?
Yeah, and probably just first look then at box number 3
Okay.
because we're already here.
Sure. Sure.
Okay, this one here. Okay, cool. Yes, yeah, that's good. Okay.
Oh, you can see there, one of my favorite things we keep finding down here, these little swarms of amphipods, just kind of swarming like a little flock of birds or something. They look just like the marine snow, the dust or the flocculated bacteria that we're kicking up, but if you watch them, they'll change their course and heading and just keep in that little swarm.
Yeah, good.
Ah, shoot. Maybe. Sorry, I got it. Why is it moving?
There's some currents today. You can also see the rift here is moving much more
That's
than
me.
Okay.
Something's pulling me towards the box, and so I'm having to pull
Mhm.
back. But I think I'm okay now.
Yeah, this is the newest, youngest of the experiments, I guess. Most recently deployed, but still can see that bacteria growing.
Yeah, actually quite a lot. We deployed this just the day before yesterday, I think. And you can see already it's like very, very fuzzy. It's also the hottest place. So experiment number 1 we measured 20 degrees Celsius. At number 2 we had 10 degrees. At this we had like almost 23 degrees. And you can basically see that this also has influences on how fast the bacteria grow.
Is it possible that by putting the box over it and keeping all that warm water in could negatively affect the rift?
So during the box, I think this time is like rather short, so this 3 hours they should be fine with it. But what you have here in the box is that the net gets clogged, and then you could get inside higher concentrations, hotter temperatures, higher sulfur concentrations. So actually, this could like influence the animals that are there. So it could be that they are negatively affected by that, but they were colored anyway on the, in the beginning. So it would not, for the experiment, it would not matter too much, or it actually doesn't matter. But but we also now so the first thought was that we recover them after like 10 days, but we see now that actually they are starting to clog already. So we will start the recovery already tomorrow with box number 1. So we kind of shifted a bit around the times.
Yeah, this was a question that a lot of people had when we were putting them down. Is it not to this degree or this aspect of it, but they were like, wait, if you're putting a box over the venting water, won't it just blow off or build up pressure? And it was like, no, there's a mesh, but now the mesh is being covered.
Yeah, so like it will never like be blown up because the vent fluid can still go through. So it's like not as dense, and also we put it on in diffuse flow areas.
Yeah.
So I'm not worried about that, but the conditions would get like worse and worse in there. Basically.
Sure, yeah. It's getting so covered with the bacteria.
Okay, can you take a frame up?
Yeah, it'll be interesting to see when these get lifted off. But now you know, if y'all ever need samples of the bacteria, you've got a good way to collect them.
Stefan is already looking forward.
Yeah. It's a lot.
I'm only thinking about how to get them best to the surface without dislodging them too much.
Yeah.
Gesundheit.
I'll get this a little closer.
The crab is feeding the bacteria. No, probably no. Can we get highlights?
This is an experimental.
This is number 3, yes.
Yeah.
Yeah, part of the collection of these will be piercing the side and sampling the fluids inside, just for anything that hasn't necessarily settled out before removing the box, but I assume we're just kind of going to put the box on the porch when we do bring it up, or I don't know that we have a particular container for it or anything like that.
Need a giant Ziploc bag.
It'll just go on the porch.
Yeah. Oh, right, yeah. So sample the water, lift the box, that goes on the porch, and then we have to try and get the rock underneath or some samples from that as well. Yeah. Yep, yeah.
Almost.
Almost.
Yeah, it's coming out of the box. From below.
Sorry?
Yeah, it's from below the box.
Yeah.
The tube.
It's like so we sealed like the crack, but it was sitting like behind the crack.
Okay.
So, like it's like how to say. So I can show you the picture.
Yeah.
But it's just like on the edge, and like the tube is of course like sitting.
Still looks healthy.
Yeah.
When we set that box, there was a rifty here and a rifty here, and that little tiny gap is
There.
right where we put the box. So that's just right on the edge.
About that power, which is pretty cool.
That goes faster than I would have expected.
There's one of
That's crazy.
the
Okay.
limpets
Can you take the thing?
that we spoke about earlier. So the limpets can feed on the bacteria here, and then they will leave little grazing trails occasionally when they do that. If you want to sort of picture them like a grazing animal, they have a radula, like many mollusks. And that radula it's a rasped tongue. Think about sort of how like a cat's tongue is rough. The limpet or chitons as well will do this, and snails and slugs frequently will use a radula to sort of grind whatever they're eating off of the surface it's on. And then many cephalopods also have radula, although some have more or less developed radula.
It's number 2.
Can I mean, once again, like all these bright lights, we'll see a lot of time, we gotta dial it down. We will lose detail here, and we'll just blow that light. But we're not doing a highlight video, so it's super critical. You can play with it, or you can play with it and see what's next. That idea I gave you of like just leaving it even a little off is only for our video, right? So if I'm doing a panorama video, but if it's like if I'm being still, more likely it's just going to be an image. You know, it's like if you wanted to play with it, it's less critical unless there's something like that.
Giant anemones, like
Exactly. Yeah, then you would want a video rather than just stills.
that's good as that.
Okay, you can take it.
Flames on the side too.
You can see how they.
Does it seem like there's more on this side than on the other?
Yeah, but the current is pushing it probably
current
like
is
yeah,
coming this way.
yeah. So the vent fluid would be pushed more towards this direction. You can also see that's like rising. So it's also, you know, it's crazy, huh? Yeah, you can also like see here it's already, it's growing out.
Yeah, comments here about the radula on some mollusks being quite strong. And they can actually be a bit of somewhat of a nuisance. Some of the larger snails and the like, or chitons especially, they like to eat algae. And so if you have an aquarium, and that aquarium has acrylic, they can actually etch that acrylic as they're cleaning algae off. So you want to try and beat them to the punch and make sure you keep that algae under control, or they will use that radula, which can be very sharp, and it is abrasive by its nature. So fascinating little critters, and I just love kind of thinking of them grazing the plains of bacteria on the side of this box, and we have seen some little trails left behind by them where they have grazed it clean in this rather thick bacterial growth.
If one day you have more or less free time, I would like to have some zombie links to collect in the way just to, I know, to have some samples for genome sequence eventually.
Collect optimal together before.
Sure, yeah. There's no Zabelid genome published.
No?
No.
Oh. Also not from shallow waters, or?
Sorry?
Also not shallow waters?
I don't think there is.
Ah, interesting. Because they're very abundant, like not only here but also like.
You can see there a bit of the silicon seal that was on the box itself. That clear one was extruded out a bit, and then you might have also noticed these little coils and blobs of a black sealant, and that was actually applied by the ROV pilots using large syringes. And that was used to sort of apply caulking to the cracks that cross underneath the footprints of the box.
There's bacteria. ¿Dónde? Was easier. Yes. This is mesh box.
Number one.
Number one.
I would almost argue that this has less growth than box 2.
Yeah,
being the same size.
height of the. Oops.
Zach wants a clip of that.
We will also like measure temperature like when we recover. I'm also interested to see if there's any change in temperature because these systems can also be very dynamic. So.
Yeah, we broke the edge off that one. That one was hard. We really think on that. Exactly.
Those two goals are seeing.
Box number 1. And I think we recover tomorrow. Yeah. Yeah.
I wrote it over.
Yeah.
Probably. Otherwise, it would be full of cheesecake.
Yes.
So
Yeah.
come back tomorrow.
So it was, I think, the right decision we made yesterday. Confirmed today.
So with the,
The frame is down there, no?
The frame is down there.
Down there.
Mhm.
Crusher.
Okay. So, Stefan?
Yes.
Now we try out the crusher, I guess.
Mm-hmm.
I would suggest that we fly up a little bit and we probably can go down because I would also like to be more on the top.
You want to measure the chemistry next, right?
No, so yeah, you can do this also.
Maybe
we're
because,
close by.
yeah, I think that would be good to do next. So we're gonna, set up to go here and find the warmest spot.
What's the program?
And then once we identify that, we would also sample fluid.
Come back.
Yeah, we can look a little bit. Little shimmering somewhere here. If you
Yeah,
zoom in
I
here.
think it's here. There's some shimmering here.
Oh, I see it.
All right, for anyone joining us, we are back down on the bottom with the remotely operated vehicle Sebastian, 2.5 kilometers back down to the East Pacific Rise site north of Tika Spire. We're in what we're calling the suburbs of Favoflo, where the 3 experimental mesh boxes have been deployed as crews. Still pretty early in the dive yet. I think we've got about 6 more hours max on bottom time. But again, if we achieve all of our objectives, we've been bringing up a little early. Kind of depends. Yesterday we stayed until max time. I've also been able to bring you the ascents recently, which is nice.
No, it's not here. It's like here.
Going to do a bit of probing here. We just had a bit of a watch change amongst pilots here, and we're also approaching lunch. We've got about an hour before lunch starts up here, so you'll hear folks come in and out throughout, I'm sure.
Temperature measurement starts at the experiment.
This for the weeks. Let me check what we wrote yesterday.
So one of our marine techs, Juliana, actually updated the heat overlay on y'all's screen right now. So the max temperature should be within the past minute now. So the larger upper number is more of a live view, and then the high temp probe max will be the highest temperature recorded in the past minute.
Yeah. Yeah, stay there for a moment. I think that's a good spot. Yeah.
Yesterday was.
Let's wait. Usually it takes a time to stabilize. That's a nice spot.
Well, that's not. Some photo, vehicles, Subseafloor, there we go. Okay. Where? 50. And then we go to the CEO.
Yeah, I think so. If we remember the spot, I mean, no, we have the temp. No, we can sample the water right away as well, right?
Yeah.
The same spot. Yeah. Okay.
And then the 50.
Let's see. It was a little higher.
Right.
I think we will temperature ultra site too.
Yeah, this is it, it's 13, 15,
The maximum was, I think, around 19.
17.
No.
Yeah.
Yeah, I think over there is probably the best.
What is
I think the warm spot was maybe slightly over here.
It's going up,
Yeah.
Oh. We got it.
Yeah, try to stay there. See how high it goes. Yeah, it's a nice spot.
Yeah.
Try to stay right there.
Now it's 20.
Okay, let's start the pump
That's the UXGX.
for the small bag.
Okay.
So Andre, we're starting pumping now.
Okay, so what should I write in the
Yeah.
condition text?
read it in the before
Drop
I
it, yeah,
change this.
so
Okay.
Can't fix marks.
it's dropping.
And we're gonna pump for like 15 minutes.
Temperature is dropping, but we're in the exact same spot, so.
Yeah,
That's where it is.
that might be part of this pumping because it sucks in some seawater potentially too.
Look in the bag. See. All right, we're gonna try and collect some water from here. So if you've been on for some of the other cruises, or excuse me, some of the other dives this cruise, we have a bag that we're going to pump the water into. Actually, a couple bags on board for this one, I think. Or is it just... Yeah. 2 bags.
So we were taking water from there. 74. This is definitely an experiment.
No, right out of the water, because it's just
Yeah, sample type order, sample type order, initial text, site. What does this say?
what the text is.
Cover flow. Cover flow.
Yeah, right, like just the text is saying.
Yeah, experiments, sample order, and is 2 grams. 2 grams for location of another.
This is just
Yeah. This is that. Something.
Can we take a look at the bag for a moment? I think it's a little hard to see if it's in place. I mean, it holds 10 liters, and we're aiming for like a few liters at most.
So the bag that you all are familiar with is this bag. That's not what we're pumping into right now. We're got another bag here.
Yeah, it's a little hard to tell. But I think
Secured underneath there. And that is being collected through a wand that is in parallel with the temperature probe right now.
And as you can see, the temperature is varying quite a bit, so that's part of this very dynamic environment. So the water comes out and mixes with the seawater, but then it's there's some flow and some current, so it always moves a little bit where the warmest temperature actually is. So we can see that it's quite variable. And the animals, of course, that live there have to sort of adapt to that kind of dynamic environment. But sometimes you might have seen that you have these very small amphipods, like little crustaceans that actually seem to hover around the vent, and then they really map where the fluid is coming out and track it quite well.
Beyond that, are there experiments that have used, say, dyes in the flows themselves to kind of see what the structure of that flow is, to actually dye it with a fluorescent dye or something?
Yeah, I mean, sometimes not. I'm not sure whether that has been done, but I think what you can see sometimes, like the shimmering water, gives kind of a very good indication where it comes out, and you see how it's like moving around a little bit depending on the flow.
For sure.
But measuring flow has been quite a challenge actually in general in our research field. Yeah, so it's not easy, especially if it's like like here we have what's called diffuse flow. So it's going through the cracks and pores of the crust and coming out at different places.
Yeah.
And so it's not always easy to measure how much water is actually coming out and at what speed. But it seems like the bag is inflating, but I'm not sure. But also coming back to the mesh boxes and the biofilm growth that we actually see on there, that's a testament also to the high productivity that we have at these systems. And all life that you see here ultimately depends on these types of microbes that grow utilizing the chemical energy from oxidizing a reduced compound. In this case, it's mostly sulfide with an oxidant that's mostly oxygen, but it can also be nitrate. And these organisms generate energy from that and use that to convert carbon dioxide into biomass. So they are at the base of the food chain of this system, which is based on chemosynthesis, as we call it. So these organisms are called autotrophic organisms, so they can create their own food, so to say, from CO2. And like you have in the surface ocean, where you have photosynthesis with algae, producing or fixing CO2, you have organisms down here that use chemosynthesis to do this process. And they are really at the base and responsible for all the productivity that we see down here. Either organisms graze directly on them, or they also live as symbionts, as we have heard in Riftia and the mussels, for example.
Right.
And as we talk later, or as we have mentioned already before, there's also a substantial subseafloor biosphere at these vents. So you see a lot of animals here and activity on the seafloor, but that's only half of the story, and a lot of growth and productivity actually happens below the seafloor, mostly again by microbes that live in this mixing zone of seawater mixing with hydrothermal fluid, but potentially also some other organisms that use that actually as a highway or an avenue to recruit these vents. And that's what these experiments are actually testing, if animals actually are using the subseafloor biosphere as an avenue to recolonize and recruit to these vents. Just a few more minutes before we wrap up. What's the temperature right now? It's not showing right now. Oh, okay. Yeah. So now the temperature is actually back to 16, almost 17 degrees, which is pretty warm. But it really, yeah, it's quite dynamic. And it's like almost like in a forest where trees and plants try to get the best spot to have enough light. So here, the animals like the riftia, for example, and the mussels are trying to find the best spot to get the most sulfide, for example, or oxygen, depending on their tolerances. So the riftia usually can tolerate higher sulfide than the mussels, for example. But that's basically, prime real estate, and they try to find the best spot for them to utilize as well. And so that's why you see sometimes also the riftia, in bend shape, for example, trying to relocate maybe to some extent. But they're, the riftia are basically sessile organisms, but the mussel can actually move around and find a better spot. Okay, that's it. So we are finishing the water sampling.
And it was like a little made area.
Pumps off. Okay, and then we can store the wand. Monica, so next would be the testing of the rock.
Yes.
You want to direct that?
Yeah,
Yeah?
yeah.
I think that's probably better than if I try to do that.
Sizzle or whatever rock thing, I don't know where it lays around. You can find this also.
You can see all the satellites here, and then they're all counting. They all at the same time. They're so cool.
So funny. Okay. Let's see. I'm gonna try to do this. Must you know what that goes in? Yes. Okay. It is. Sie tun ja noch zurückkommen, ich weiß es nicht, weil das nicht das liegt da, wo der Rockbreaker liegt. Okay, so I'm not sure if it's on this or the other side, but if we go a bit higher, then I know which side.
Das ist gut, weil ich muss schauen, weil ich will, dass es möglichst weit oben, aber natürlich nicht neben dir.
Ja.
Das ist gut, das oben drin, das soll Wasser fallen. Rock crushing test number 2. Can you make an observation just that with the highlight frame grip, the
Observation or highlight?
no, yeah, highlight frame grip and write down the two worm clump next to box. Which box is this?
This is box 1. Oh, no. Okay.
Yeah, so box 1, next to box 1 is already, we did already the temperature of this, so we're not doing it tomorrow. If you can fly up a little bit, I look at the area and also for the rock, also for the big sizzle, you know, this big thing.
Oh, right.
Yeah, we will try to find a place for crushing with the big thing and the small sizzle first and then the big, maybe something like this. But ideally would be a high up place. Just see.
So it's either on this side or like really opposite.
Yeah, here it stops, yeah, no? I mean, the side is good, but the on the, you know.
Okay, so if you turn, look in this direction. It's very close, so we should see it like in other directions.
Yeah,
A little bit down.
you can take this here if you see it.
Yeah, so here is the weight from the
This
others.
is the weight we need to get
Then
also.
further down.
Yeah.
See about south.
Uh-huh.
But this is not what we need. It's like the
The big bar.
The big bar, yeah.
yeah.
Big bar's right there. See it?
Yeah.
So land. See.
And here's the rock crusher.
Yeah.
Yeah. I would, if you look in this direction, then later we follow this in this direction because we are high up. We just see what it is, what we can do. Maybe like over there, like it follows here. See over there, maybe something is higher.
Yeah.
Because here it stops with that big clump.
Yeah.
And
So we grab us in
yeah,
He also wants the rock crusher or only the chisel.
well, I want
It's
to
not super successful.
this chisel. We try, also if we let's take the bar. We have the little one. I would try with the little one some first and then go in with the big one. And I want to see if I do it on both sides. How we can approach this as if we would do it tomorrow. Das ist genau schon noch etwas. Alles liegt, das ist drin, wir können die aber noch nicht mehr. Okay. Okay.
Okay, so you don't want to do anything here then?
No, not here, no.
Okay.
If you
Yep.
this will take long. This will take long and the school experience I can video also so you can for sure go for lunch when you run them.
Okay.
I can just do things because I know already.
Then
And then you take over for the extra whatever.
or I can do the slurping on the bishop's fire. Slurping on the bishop's fire. So we do the school experiments,
Yeah, I go.
and then you also
Yeah, I do
look
the
at
school
max true.
school with labs. And then you take the video, then you can take over for slurping, for extra.
Yeah. Yes.
Event.
Yeah. Then we can also check event and biomass. Okay.
Yeah, yeah. Because then you have to break and can eat and then.
I'm still curious about
Yeah.
this.
Yeah.
But thanks, yeah, no, that's. But I can also do you and then get lunch too, so I can come back.
Yeah, with this, I want to make this
I
and
make it quick and then come back.
this cool experiment. Let's see. We will see how long it takes because yeah, at a certain time.
Right?
Yeah, for the bag, for the blue bag, you need to not if you just walk in, but I told him it takes long enough. For the blue bag, you need
30
to
minutes.
So he has to come in. You need to get him for the blue bag for the switching on and off here because he knows most about
Okay,
it.
yeah, that's
It's
good
the
idea.
easiest
Yeah.
because
Yes.
you're balancing this. Mikado. It's like this Mikado game, no?
That's that.
Okay. So I was thinking, where's the? This is the big one. that we go up here and look in this direction. So we look, where are we now? Where's my... Where's this? Oh yeah, here it is. Okay, we go over here. How far is this? Over here would be. This looks good, but this is easy. Maybe. Is this far away? This should be far away, no? Because this one is easy. I can see you can just flip it to this side. Yeah, so here, like this here. Do you see how far this is?
Look on this side there, looking down.
The other direction. This is one. Yeah, but this is good, I think. Or the other ones. Yeah, this is. I just look if this is far away, but I think it is. Yeah.
It's fairly far away. So
Whether you sit on, either from this or from the other direction, I don't care. But it should be like maybe here somewhere. Yeah.
so down here.
For this one, so the not yeah.
That is Dr. Monica Bright working with the pilots to find the best place to set the sub down.
This one, a smaller one. Okay.
It's another side of that.
Yeah, I think this slope where you are right now is steeper than the other side. I think, but. Yeah. Where is this? Is this completely steep or? Okay, good.
So that's the box. You can see the boxes off the both side there in this camera.
Yeah.
So I don't want to get too much. Don't want to get too close.
Can you look on it?
Can I look here?
Yeah.
Yeah, this is okay. If you go more, if you sit down more here. Huh? Yeah. Highlight, no?
Nice view.
Yeah.
It's pretty close, huh?
Is this close? This is not so
It's
close.
like 3 meters away. You might guess.
Take this one here. This was the one, if you see a bit this.
So we've set down on the seafloor here. Got a nice line of riftia leading to
This one.
previous
possible?
experiment dye boxes.
Yeah, no, I want to. Can I look?
Yeah.
Okay, let's look.
Dr. Bright's pointing out some samples she'd like to gather here.
Let me look first over there. Where are we? Where's the box?
The box is right there.
Right there.
It's a little bit dark.
Okay, I see it. Okay.
Estimate the distance, like 2 meters, 3 meters?
Looks like there's a bit of a slope. It's probably seafloor, so bouncing on that port ram.
When it gets not so far away.
3 meters. Man, what could happen? Also, let's think what
Stuff
could happen.
falls down and then there's a lot of movement. If we have to pull back the rock or whatever, then it's, I don't know. I find it a bit risky, to be honest. As a first tryout with the mega chisel.
Let me just look. This is a further away, no? Here we are further away also, no? But
Well, I can't really land on that
Yeah.
because there's a kind of mound.
let me see. Also, this is the wrong direction. So what do we guess is the distance between this and this?
Like say some 2 meters approximately, maybe a bit more, not much.
Hmm.
How long is that
Hmm.
stick?
1 meters? Well, not 1 meter for sure, 1 meter.
Let's go up and look a little bit around if we find something further away. This would be good to test because I think it's very similar, but I have no idea. Okay. Because this is exactly this ridge,
Yeah.
and then it goes down, and then you cannot sample anymore. And the other ridges might behave completely different. No, there is no. This is the ridge where they come
Yeah,
out.
very
No,
fast.
so let me see. Also, I'm not. Can I use the camera? Also the.
Yeah.
Let me just see. This is the. It follows here, no, but it is. It goes down. It is super steep, so this is like. And then we go over there. To this. We can go maybe like here, but this is all muscle, so I don't know.
What about, because before we have been like sitting here in front. This was where we've been sitting in front. What about if we go a bit, it's a bit further away?
Yeah,
that,
this
that
is-
would be fine. Or like.
But
one of
here is
these
also
here.
good,
Here,
maybe.
probably.
This here, yeah. Or this here. I mean, this is smaller. I mean, this
Yeah.
from
This
this
one,
side
yeah.
we are away from.
Far
Basically,
enough
Yeah, I
away.
can try something, yeah.
Here?
Just there's a ridge there, so I can
Yeah,
have
yeah.
to go.
The scientists work out the best spot to land. We'll do a quick recap here. So this is the Underworld of Hydrothermal Vents expedition. This is live footage from ROV Sebastian of the Pacific Ocean, and we're on the East Pacific Rise. And one of the main ideas of this expedition is to study whether organisms can be transported through the hydrothermal vent system below the seafloor, or a bit like a underwater subway. So I'm doing a range of experiments, and here today it looks like we're gonna settle in a good spot and maybe do some more sampling. We've got Dr. Monica Bright, the principal investigator. You'll hear her on the science microphone. Got her microphone.
sorry. Well, so we try to go with the small first here from this side, and then
Small chisel.
small chisel right in here and try to move and maybe go in with the big one first. Or is it? Or do we try the big
I
one?
give it a go, but the rest of it's heavy, so it should. There's a gap there. It'll go through it.
Maybe try with the small. The idea would be, also my idea is, I don't know if it works at all, but my idea is to try to get something off here and then do the same on the other side of this little clump and work our way there and then try to lift something. So the goal would be to get the rock in the crack right where the tube worms are. Maybe with the tube worms on or so, to break it so that the tube worms either fall out or stay on the rock. And this is the side I would like to look at, theoretically.
And the purpose is that we want to figure out how we can recover the rocks for
Tomorrow,
experiments. It's a tryout.
This is a trial for tomorrow. Because our experiment.
It sounds like we're trialing a rock sampling process here. Going to do a little seafloor surgery with the chisel.
Break, also.
So we can start like in between those two clumps.
Yeah, like. With the goal to touch. Okay. Yeah, this one is more visible, this crack.
But if you go closer to the large tubes, I think it's easier.
Yeah.
Because there the crack must be larger.
Like somewhere here. Here you can see it, but it should be also. Yeah, I think you're right in there now. This is good. This is good. maybe it's possible, or maybe it's easier first with the smaller one and open the hole a little bit and then with the large one, I don't know.
I'm gonna approach it. Is it here? Das ist nicht yeah, so what I did, come back. Hey, good morning.
So the pilots are working on ground underneath the base of the manipulator, so difficult angle to grab. And they shifted the sub around a little bit so they could bring the porch all the way out to get a bit better view, and hopefully a better angle to grab that tool. Because often where the sub is landed out, so now they'll bring the starboard manip around. when using the manips this close in on the sub. Pilots are constantly aware of items they have in the porch, like that big blue cooler, different cameras. Right.
The measures to break?
No, we're
How do you pronounce it? Chisel? Chisel?
chisel.
Alright, now it chisels out. We'll do a handoff. Get it back in the port manipulator, which has a better grip on that tool. You can see Chris has fitted the chisel right in that T-shaped indentation on the port manip. You can see on the starboard manipulator and the jaws, they have a bit of a soft squeeze in the core. Now the pilots will reposition back to the original spot and get to work on that crack.
Right, so that was the one you worked on. See that? See the end of the bars? The crack there?
Yeah.
Plug the muscle.
Where is here? Right here. It can be from here to here somewhere.
Could you try to go with the chisel in here?
Because
Have
if you
the bandits one in this direction, like this.
yeah, or
But
over
you cannot
here.
get in. I think there's no way around to
Yeah, then go here
to a large one.
to the here. Right. Start here, maybe from here.
I think you really have to follow like the large tubes, otherwise you're not getting it. want to
Ja,
see
ich will nicht was Leichtes probieren, ich will das probieren, was gleich ausschaut. Mhm.
I think you've got it on you, that's not one.
Should we grab this thing?
Yeah.
It looks like the experimental site, and if it's easy, then not. Also this here,
Yeah.
and then see if you can.
We're gonna have to re-grab this anyway, so it's a good time for us to grab that chisel and the other arm. Now,
That's okay if I reset. Which one do you want to pull out?
just lift it up, I'll see. Just a tiny bit of it. Right, just lift that up.
Grab here.
It's,
What's that?
yeah, yeah. Yeah, you can lay it aside somewhere. I think here, you see the crack here. I mean, just, you can also ignore this and just try to go in here. Yeah, if you get it out, good.
Crab is matching.
Yeah, yeah.
really nice pork. Mashed potatoes are great. You know, vegetables, salads good for me. Fresh rolls, which have meat.
All right.
Pork's quite nice.
Oh, really?
I'm trying to just
Okay.
to pry up this rock. Is that what we're doing here?
Okay. We're trying to get from this side
Yeah.
and this side the rock loose so we can collect everything that is basically in the crack here. Loosen it. I don't know if this is basically just
I saw the backside around there.
Did you?
yeah, I saw it also.
Do you need me to move closer or anything?
Yeah.
Let's get some tea.
Move closer up, we grab it better.
There we go, hopping closer.
Tap it to the right as well.
Down, down. Tap it to the right, you say.
Way you get on your forward, just more right. I'm using the forearm.
Is that for you? More? Less?
I'll back up.
Back up a little bit?
You have to check for the other boxes also there, I think, behind.
Right there.
You said the other
Okay.
the boxes are behind?
Hmm.
You said the other boxes are behind?
Yeah,
Do we need to check that before we land out anywhere, or?
we checked it out, yeah. That it's good, but
But it turns count minus one alarm somehow too. Weird. Flip around, I guess.
What is the Z and this timestamp stands for?
Let's see the timestamp.
Yeah.
Yeah, it's just a time that's set for that.
Is that for what?
Can't remember.
Okay.
Where are the messages?
I'm just trying to, because that makes sense, is the date, and then this is the time, but then there's the Z.
Yeah,
What is the
that's
Z?
for just the end of the time, really.
Okay.
Yeah. Kind of like a carriage return.
Yeah.
Checksum.
Yeah.
Hold on.
You want to pass it to the server? Grab it. Yeah. I can do that one.
We'll do starboard.
Got it?
Yeah, good.
Okay. Increasing, opening. Going.
How's it going? Any?
Yeah, just try from this side where the ones are there already, or from the other side we take over to touch it. Because one was shaking already. The upper one is shaking briefly over here.
That's why I asked what's going on.
Yeah, I can put a little more. I got 42. A little more. I can't go more if I'm in cruise.
Oh.
I can't.
Oh, yeah, it was my
We're so close. That's
Yeah.
what I meant, like. Gotta have a little bit more.
Something's, hold on. Something's up with this thrust. Is that Alex? Maybe. Upstairs? Yeah, I'll get out of here. I'll settle down now.
Not much of that. Take your out process.
Yeah.
Alright.
God. Yeah.
This road is on me, right?
See it? Just a one right there? Yeah. That's where I hold it. So it's pretty close. A little bit in front of the RVA.
Move forward.
No, don't go forward. Don't go. That's a pretty good spot.
Down.
Was it here, right? Yeah.
Let me settle down a little hard this time.
Okay.
That's 52% down in cruise.
So yeah, it's pretty nothing. That piece of rock.
Or if we could put that big, or if we could put that big one in there now.
Yeah, it's really big.
Yeah, the big chisel maybe. Maybe that's enough of a gap now to actually put that in.
Almost there.
Scoot forward.
Wow, that's a huge net is there.
When I saw that whole plate move, like even if we're breaking free, I don't know what we're gonna be able to do with it. It's huge.
Yeah. Or we'll just see.
I think the hope was maybe, you know, if we lift it a little bit to actually drop a weight on it and break it that way, but I'm not sure.
I need to be able to work like in this area.
I need to scoot to the right, right? Yeah, scoot to the right.
Kind of wish I had a deeper hole. Alex, you want to jump on this arm real quick? Yeah. Try to pass me a little bit deeper grab.
I'm trying to change my angle a little bit, because otherwise I'm going to be sitting on the rock you're trying to pry up. You know what I mean?
You're gonna grab higher above.
I'll grab high, I think.
You want me to grab high? You want to grab high? Okay. Sorry. Oh. Yeah. I gotta keep it.
Okay.
Oh.
Alright, glad I can help.
Settle. Put you down again. There's your crack right there.
Okay. Yeah, I mean.
Do the bend.
Alright.
Do the bend.
Okay, one sec. Okay. So everything.
We should turn your angle more
Try to pitch it out.
that way, you know? Yeah.
So there's also a limit on how much we're going to put this on for the right. Yeah. This is a little much.
So it's difficult to get that into the crack, or?
Yeah, it's just such a big rock, I'm not really sure we can move it.
Wait, I think Sabina should be in soon, so I don't want to make this decision right now. But I mean, it's a very important part of the project to actually get the rocks once we remove the mesh box.
Yeah, that's true. But
And
can
so-
we not look around and find maybe one that is?
Yeah, I mean, the thing is, like, you know, yeah, we don't, I mean, I think this is I think what Monica tried to identify right now is basically what we expect to be below the mesh box.
Oh,
I
so
think.
this is more of a test.
Yeah, it's totally a test right now.
See how we
Yes.
dig these rocks out?
Yes. Yes. Exactly.
And the corner of the first day that was moving, have you tried in that location? The first day we managed to move, but we didn't manage to recover.
That was under the box.
It's under the box.
Yeah.
Oh, right.
That's what I'm saying. There is probably some locations where we can do this, but I don't think this is.
Yeah, like this may be down here, but I'm not sure. That's an overhang, right? Yeah.
Oh, that's what you're going for, yeah.
Just picking it up so we move it out, you know?
Yeah, it's totally a test because we are planning to start the recovery of the mesh box tomorrow. So that's when it becomes important to actually recover. So with the small one, we're able to lift it a little bit. And then, so we then got the one with the big one, but that's really difficult to get in there. So the question is now, and they're also mentioning that it's very tough on the arms. That's what we're doing right now.
Yeah.
That's something to consider as well.
Okay. Yeah. Okay.
But of course, it's super important.
There's some boxes.
So that's what it looks like.
Yeah, I'm pushing so hard I can't even keep the bar in the grid. It just bends it. It's moving the RV around. That rock is very large. We did manage to tilt it, but the rock it's like a meter by a meter. It's
Okay.
huge. I don't know that there's any way to do that.
Yeah. Can you try once more that I can see it? How it's moving, the rock?
With the small,
Or is it
Trying
not?
to keep, but
But
this is a lot of abuse. On
yeah, yeah. Okay.
At a certain point, you happy with what we're doing with the arm?
Yeah, so far, but, obviously we don't want to beat it up and break it.
Yeah.
maybe in the end, like the best, something that could be tested is like to just try to chip the surface with a chisel and see how that works.
Gently.
The third box is where with respect to
They're all in front of us now.
So, did you have automating out for a reason?
I just didn't want extra thrust when I didn't need it.
Yeah.
But you can turn around if you want. Seem to be holding it to 50%.
Well, we were all the way on the other side. It's great. It's not appreciated.
Yeah. The third box is more to your left. You can see it in that camera.
That's okay. First, I only see two boxes.
You're over the side that we're trying to work on now.
Okay, so come back, slide back a little bit.
Slide back and turn your heading left.
So go left.
Just heading.
Yeah. So it's basically this plate here that started to move, yes?
Yes.
I thought it was the one on the other side. That was
Yeah,
Yeah,
This
it's
one.
the far side.
Okay, so it was that one.
that one started to move here.
Okay.
But we only ever saw it move with the small chisel. They haven't moved with the large chisel yet.
Yeah, because we were not
The large
able-
chisel's too big to get in.
To get in, yeah. Then
Crack
you can
there,
crack.
no?
Yeah. Yeah, then, yeah, no. But it's not, it will not go further down, no.
Yeah, it's not. You don't really get a good level.
Could we try to pull these tubes out?
Yeah, just put them in it. Just pull them out. There was one.
Yeah,
Pull them
so
out from
pull them out to see like if if this helps, because there would be a larger crack
Oh, like
here.
maybe there's a hole there
Yeah.
or something?
Yeah.
Okay. But we're still working with a 1 meter by 1 meter square
Yeah.
of rock.
Yeah. Okay.
So if we get in there better, we're still like this is
Yeah, this is like too big
rock
to. Yeah.
manipulate the jar.
Yeah. Okay. Good. What was Monica seeing at the end? Did Monica see that or?
No, I think she saw like that the plate, that the big plate was moving, the small
Okay.
chisel.
Mm-hmm.
That's when I was kind of joining here.
Is it possible to get in with the small chisel
Under there?
into there, and when it's like lifted a bit, keep the chisel in there, and then throw this
Yeah,
the
that's
rock
what
cracker on top?
I mentioned that earlier.
Because then it's like the plate is a bit like lifted up, has a different angle.
We can try. You want me to give it
I don't
a
know
try?
if this
Yeah,
would.
go for it.
Yeah.
Just so much needs it.
What's that?
Just so much need.
Alright, so.
Just some more, cross section. That's it. Just bump those open right here. Just double pass it here. That. Yeah. Freeze.
We've got Zach co-piloting right now, and he's using both arms to get that small chisel in the perfect spot to grip it with the port manipulator.
seems much easier to start with this small chisel.
Okay. Yeah. Mm-hmm.
It's so big.
Yeah.
Trying to jam it under there, but
If you get it, you just like to push it on. That'd be easier, then just so you don't get that extra. This is road, part of the
that's not me, that's not me.
Yeah, no, that's good. That's one reason I'm just. I can hop back up later.
Just go a little bit.
This rock is too big, too heavy, also for the arms. But the plate can be lifted up, it's moving, but it's too big, and like then there's also too much tension on the arms. So we also do not want. So we're trying right now if it's possible to put the chisel inside, let it fall on the plate.
Die anderen Zimmer. Ich glaube, nicht besser so in den Schottischen zusammenbringen. Mhm. Nicht. Ich meine, vielleicht sind die morgen nicht Cracks, ich weiß, aber ich meine,
Morgen sind mehr Cracks.
Ja, man kann nicht das ganz leichteste probieren, weil man muss halt trotzdem probieren. Wie schon mal, es bewegt sich.
Ja.
I can't push it in anymore, and if I let go of my fault, I'll try it. I'm not sure what else to do with it. Instead of freezing, unfreezing, opening.
No.
Okay.
So my hope for tomorrow is because tomorrow we have a bit more cracks.
Yeah.
We usually have like not. Yeah, we have more cracks in the experiments, so that probably. I hope that the rocks are smaller, that could be lifted up a bit. That's the only hope that I have at this moment, because this is not possible, I think.
Yeah. The big bar is not so good because it does not fit in here good, no?
Yeah.
Okay. I mean, I'm just.
If you stop the chiseling,
This is quite a big rock, you know?
Also, it's
yeah,
a big rock, yeah.
So it's hard to do anything.
Yeah.
But what we could see is that the rocks can be lifted up, and I think if under the experiment there are more cracks,
Yeah.
looks a bit different.
Yeah, it is difficult to find a place where it's exactly the same. Maybe possible, what not. So it's also good to try.
Experiment. The team wants to try tomorrow.
Yeah, it's only 12 o'clock.
So as they noted, it's impossible to exactly replicate what we'll see tomorrow, what kind of sampling they want to do.
Yeah, but this is also
But they're doing their best to work through the pitfalls and possible strategies
Here we're done.
first.
1, 2, 3 places very fast. Let's say like this is. Yeah, this is the next. This is 1, 2, 3 for me. Yep, you know.
max 2 and then
This is.
reception samples.
Oh, crab spa.
Yeah, we have crab spa.
This others.
Better than the large one. Also think because it's bended a bit, that also helps.
Ich weiß gar nicht, ob das hilft, wenn es reinkommt, du kommst nämlich dann auch nicht gut rein. It's just a 10 minutes or something. This one is more something if there's any change because yeah, but I don't know. the others.
But you know, if because there are smaller, like more cracks in the experiment, so I think it could work.
Yeah.
Yeah, that's,
Yeah.
we're not gonna be able to move this one.
No, no.
Yeah, but I think we know obviously we can continue the next day a little bit. Because there are smaller places.
So what should I do for now?
We
You wanna leave this little chisel here with this big one?
We need it another time when we dive. It's always good to have a chisel.
School experiments.
I will use it stay for the
Yes, for sure, totally, yes.
But you can take over.
So after trialing that, background are 3 previous experiments that the team has set up down here on these Pacific Rise, looking to answer some questions about and if an organism is in there that has not been dyed. The idea is that it will have come through the cracks that vent fluid is seeping out of, because those boxes are covered in a mesh that should keep things from the water column from getting in there.
Side area with the children.
So the pilots right now are taking a turn out of the umbilical. We have Zach's piloting. Nope, Zach is co-piloting. Alex is piloting. The Marine Tech for this cruise. Talking to you right now. And where are we headed next, Monica?
We go to this basalt area where we have some experiments from Austrian and Slovenian schools. Those are actually the control areas because it's cold there, dark there, and lots of pressure. And in the one experiment, it's investigated whether vent animals from down there would preferentially eat meat products over plant-based products. So we made a grid and we put on the grid liver, fat and meat, and as plant-based stuff we put in chickpeas, nuts and something else which I cannot remember now. Chickpeas, nuts and what else, Salva, did we put in? Chickpeas, nuts and and a potato.
So we brought a picnic down to the seafloor to
Yes.
see what the animals prefer.
Yeah, because all those the other experiment is from a Slovenian school, just very small children, and they just want to see if the beans can also sprout in the deep sea.
Cool.
So we will see this in the same deployment. We will very soon see, because it's already in the picture, the 2 lids. These are bucket lids, so we just with the names of the children in it in the school. And the same deployment we will see just then after we will look at this at vents, so where it's warmer.
Cool.
Oh, and I
it
can
looks like
see
you got some
that
visitors down there, huh?
it attracted a lot of
Got some friends.
What's the name of the site?
This is just basalt, Tika basalt area. Whoa. And school experiment, Vienna school, from the other one is from Piran. And this is the bare basalt control, actually.
It looks like some items are definitely more popular than others
Now
on
we
that
have to-
tray, huh?
Right, yeah. We saw already
Some might already be gone, right? No?
in the vent, also we will see how the vent looks here. When we looked yesterday, did we look yesterday?
I wasn't here.
Ah.
Yeah, but I think they looked at.
There was everything there still, but now there are even more animals.
Potato doesn't have many chambers.
What, do you remember what heading you were looking at first time? It might be back. It might actually go up 38 meters.
It's where better fits, you know? Just came a little bit to the right here. Now go around that drive.
That's where it was just a second ago. I was just
Pictures and
The potato is untouched.
You want to take some frameworks?
yeah, but and you can put on a highlight video also, Salva, and then you take some. Okay, say running. Okay.
Somebody's thinking about the beans anyway.
So you can see on the grid, on the back of the grid is the chickpeas, then in the middle is the nuts, and then the potato is clear, and in the front from the right to the left. I think that, I mean, if they just sit on top of it, does not necessarily mean they eat it, because I think one squat lobster sits on the nuts, but it reaches out to the meat, also to the liver, you know. So, and they really.
There's a tiny isomer, I believe, in the liver. On the liver.
Okay, let's see. Oopsie, wrong direction.
There's a hole to the left. You can see a shrimp popping out now and then. It's off screen.
Ah, yeah. Yeah, they are also there. We can, yeah, here also. But I mean, the squat lobster sitting in the middle of the nuts, even is, I don't know, is not doing anything, because right now I think, I'm not sure.
I'm surprised there's still a lot of left.
So there is a consensus on eating animal source foods.
Yeah, I mean, the children had the idea that they should eat first what they know, and in the deep sea there are no plants, so that was the main idea.
Can you guys tell what's been picked at the most?
Yes, we can. Okay, so this is checked. Now go to the beans. Where's the beans from the school? So the sprouting bean experiment is made that the animals cannot go on board the ship, and the children did also the same experiment at their school already. There's a diversity of different beans in here.
And they are not sprouting.
Yeah. Pictures of Sarah? We have a lot of pictures, no?
Yeah, I think.
Okay, then we can go to the number 60. Yeah, I'm doing first the school experiment, then the other, but yeah, you can text him.
Yeah, I don't know if there was one properly marked.
Ah, yeah, this is good.
I see waypoint 16 experiment.
Mm-hmm.
Where is it?
Mm-hmm. It's just around the corner. Also, we
Oh,
turned.
they did a different color.
Oh, I see it.
You see it already?
Yeah.
It's going to be around Bischofsweier. I think that's where
Hmm?
we're going. That's near Bischofsweier, right?
This is all like,
Yeah.
like very close. I think here we are. We just go in from here.
This is the one that we didn't really, liver
We
was
did,
gone,
yeah, it was gone already.
but we never saw it.
We never saw it, somebody eating, but it was gone, so disappeared.
Someone stole it.
Someone stole it.
Oh, there we go.
And yeah,
So I used that camera, just orient the science camera like as centered as I can, so I'm set down like the best chance.
maybe we should have cooked it. The chickpeas came out of a can and they were cooked, no? But the potato, we just thought, okay, why not? I mean,
There's no butter, there's no sour cream, it's not I wouldn't eat it either.
yeah, but
The anemone is still there.
yeah, we go to yours right after. Just checking this first, and then we can move with the camera over there.
So the only thing that is really eaten is the liver.
The liver was gone already the last time.
The meat is smaller.
No action, but liver gone.
Yeah, the others, it's funny, because the liver's gone, but the others don't even look like touched, you know?
Yeah.
Yeah, I think it's the opposite of what I would have expected, because I thought, okay, many animals advance, food gone fast, but I think maybe it's the opposite, because when they're on the bare basalt, they take everything and they run for it, and here there's so much of other food, they prefer the natural food.
Yeah.
Where the liver is, I don't know. Is it looking here? Somebody took it, the whole thing. I'm just...
The beans are also...
The beans are there, nobody took it. Did you take a picture also? Okay. Then we can move to the next one. We have a lot of experiments here. Max, you can say what this is, yours. Where is he? Ah.
Wow.
So, what do we see? Landscape.
Is in this microphone?
So here we have a tableau of human artifacts, or you could call it the dregs of humanity. And oftentimes I treat my sculptures like events. And in this specific instance, I'm surrendering this sculpture, which is presumed to be artificial to this habitat, and letting the natives and endemic fauna and bacterial communities continue its progression and to denature and evolve and transform this my assemblage. So I guess you would say it's in my work I'm inviting a lot of what I would say is co-authorship. Fine.
And who are you?
Oh, I'm Max Hooper Schneider from Los Angeles.
Nailed it.
Cool. Thanks, Max.
So we do have an artist at sea program here on Falcor 2, and Max is our artist at sea.
You cannot see totally clearly what it is?
It reminds me, Sgt. Pepper's Lonely Hearts Club Band. Cover.
Hell yeah. See, there you go. That's like, I told whoever was interviewing, I told Monica yesterday that my
I watch it
the
anyway,
approach
but
to my work is that I offer a set of conditions with no plot. So there you go. You just finished the plot for me.
So it's the warming of
the warm waters.
And Max, you've been busy on the ship. You've been all over the place. You've been working with the engineering department to do some of your work,
Yeah,
and the ROV team. And
it's been nonstop. I mean, at first it's like a traveling incubator. So with an endless amount of resources. Yeah, I've been working with the metal fabricators and the welders, and encouraged by brilliant Monica and researchers and principal investigators. I mean, it's like a panopticon.
Can go to Bishop Spire,
You guys, you know,
which is very close by.
it's probably the most high functioning environment I've ever been.
That sounds good.
Yeah. It's weird to also, it's I've never had an existence, I'm telling you to shut up, but I've never had an existence
I think we're
that
done.
has such a verticality to it. You know, like you sleep, downstairs there's the labs and the gym, upstairs the cafeteria, but it's never had that. It's an odd one. It's like a 4-story house that's kind of gently rocking.
Yeah.
My thighs are toned from going up all the stairs.
Yeah. Let's look at the next one.
Did I invite you?
Bishop Spire, yes. I think it's very
Sounds
close.
like an album title.
Might I tweet it?
What's that?
Might I send it?
It might be back here, this one here.
Bishop Spire should be right ahead if you turn a little bit to the right.
Yeah,
I think it's
based the last couple. No, I'm gonna keep on. My job is to give you energy. The next two, I think, are gonna be actually like the superstar pieces.
There you go. This is the stable collector from
Tiers at the base, and then the whole structure is actually covered with Albinella pompeiana, Pompeii worms that form tubes and actually colonize the outside of hotter structures like these chimney-like structures. So they can actually tolerate temperatures up to like 50-60 degrees centigrade, which is quite remarkable for animals. And here again we see another piece of art by Max that has been deployed a few days ago here at the site. And maybe Max, can you talk a little bit about this as well?
This is so white.
Yeah.
Yeah, we need to work on the contrast a little bit here.
And the mic. Yeah, thank you, Stefan, for the introduction. This is probably actually a more clarified example of the first experiment, which has the specific intention of taking something that looks abiotic or artificial or even like some sort of pollutant until it's embedded in the habitat and subsumed by local element, is supposed to become bioactive. And you can see even after a 24-hour, 36-hour period, there are a lot of things that are exploring it and making nests, and it's probably slick with invisible bacteria. And I guess the long game narrative would be that we have an inevitable inheritance of pollution from centuries of wanton destruction, and we have to actually think about what's going to happen to it. So this is really supposed to show some sort of potentiality in that front.
Hey,
We need Dave Attenborough.
hey, this one, that's the photoreactor.
Photoluminescent. Yeah, there's strontium and iridium.
So would it be worth hitting that with all of our lights and then turn them off for a second?
Oh, that's a brilliant idea.
You said it was a different wavelength.
I mean, but any it could be an interesting.
All right, we'll see what happens.
I mean, it's worth a shot.
Make sure your aperture's all the way open there.
It's also worth a shot for future generations because that has those pebbles hit it.
All right, so all the lights are hitting it, and assume your aperture's all the way open, right? Very faint.
Very faint. Very faint.
The lights off on
Crank the
But it's
ISO a little bit.
But if we did a long exposure at some point,
If we crank the ISO, it might work.
Yeah. Also, the manipulator light is so well.
It's an interesting concept we got going here, though.
All right, hold that camera view. Where's moving at? One more go here with high ISO.
There we go. Interesting.
Oh,
Yes.
cool. Super cool.
Yeah.
Wow. Wow.
What a
So that's cool.
It's a very cool.
This is.
We should see if we can get a long exposure shot at some point if we can. But.
All right. Cool deal.
Good call. Good call. You know, I mean, this is a perfect example of just how nutritious the whole team is. The nutritious collective. Everybody's exchanging ideas, giving one another energy.
I will never not.
If you had a sourpuss or someone that.
Yeah, in the Upworld.
I'll never not be able to think about this nutritious now.
I know.
It's
That's a strong label, Max.
Intellectually nutritious. Yeah?
I love it.
Just working around.
Thanks, I love it.
Do you have a title for
Look at the
Not yet.
worms back there, little scale worms, huh?
Yeah.
I mean, the deployment series that will have to go all over the world together.
All right. One down, Michael.
Do you think we can just do one more, like you know how yesterday we were kind of doing a super zoom?
Yeah, I tried to do that a little bit, but I can.
I think I was talking, so I probably wasn't.
Yeah, yeah, we were doing this a little bit.
1, 2, 3, 4. Yes.
You got a lot more stuff to do? I'm out
I'm
then.
just counting the. No, I think we have time. Because we have still. More than 3 hours.
That's a good comment. I wonder if that's the first, photoluminescent deployment in the deep.
That's a good question.
Or this environment. I'm sure people have done something.
It's interesting too, because once we leave, it's going to glow for a little while after.
Yeah.
It's like
And to them, that's probably going to be really bright. To the creatures that live down here.
They're going to be so amped.
It's like the sun.
It's like an ephemeral discotheque.
Yeah, right.
We're just
very time sensitive discotheque.
It's a daily chore to try not to anthropomorphize everything.
Sure.
The pouting eel.
Yeah.
Oh, so sad and curious and lazy. I love that.
Mhm.
That's cool.
Okay.
Yeah, awesome. Thank you. I appreciate it, guys.
We're actually staying here. We're going to do a couple of slurp samples.
Right.
And so the first one will actually be with a very fine mesh.
Thank you.
going into number 5.
I got lessons. I'm gonna fly off so you can better. I'm gonna have to fly off because I think
We can probably slope like a little bit above the structure here.
There you are. Alright. So it's just kind of falling off from down there.
you're gonna just hop through my navigation.
I'll give it a whirl.
So we're going to do next the slurp section sample, and that's going to go in
Oh, I got you right over there.
chamber number 5. And I think that's going to be sample number 70,
Well, look down for
So
soup.
you told me jar 5,
74, I think, right? The next sample?
then 75 I have.
5, okay.
So this is suction sample jar 5.
Yes.
This is a large mesh,
No, it's very fine mesh.
very fine. And we're still in Tika.
Yeah, at Bishop's Spire.
Bishops.
So yeah, let me know once you are stable.
Oh, it has to be
Yeah.
grabbed like that.
Yeah, okay, I see. Right.
Certain as it's kind of falling into a weird spot.
Oh, I see. Okay.
Yeah, so I can't be laying it out.
Yeah.
My cooler pretty well. Oh, and now you got the intent probe on the back. Okay.
And also just following up of about the art that Max has brought here and created. It has been also like a very inspiring and creative process having him here on board. It's like a really good synergy between science and art, and we all have been.
So our starboard manipulator has a camera on it, so we got that view up in the picture-in-picture, although it's
What's that? Yeah. It's not right thing scared. Good job. Good job.
Crash. Good job. I know. Well, I gotta make that myself up on my science game because I got nothing on it's a camera. See where I'm at.
Cool. Jeez.
Water near flow at the base of big waves, small gap.
Oh, yeah, okay, that's true. Okay.
Basically, I've lost vision of where which is not a big deal.
this action is taking place in front of the porch, out of reach of our science cam. Occasionally the suction sampler will get loose and dang a little bit.
Let me see if I can. Go ahead, Rick.
Can you come back on that a little louder?
This green, the green sea screen is
Yeah, did you guys do any changes to the HC scheme? Down on most of the waypoints and stuff on that?
Negative. We haven't done anything down here. I can try and push them for you again. All right, so you don't have the Falcor waypoint or any of the other ones, huh?
No, I think
All right,
back
let me see
there
what I
there's
do.
a Bishop's Spire probably. Down here.
Sorry, I wasn't watching.
Bridge RV, did that do anything for you?
Yeah, I got a bit of black.
Merry Christmas.
Yeah, high enough with the nozzle to land out on this.
Yeah, so we can probably sample up here, so not too close to the, or on the side actually even, so we don't have to necessarily go to the arc piece.
Yeah, like a state.
Yes.
Should we well high of everything?
So this is Bishop's Spire?
Yes. Oh, Stefan?
More superficial.
Yeah.
Okay, and where are we sampling?
So I think, yeah, a little like further up here to not interfere too much with the sculpture.
Well, hold on, let me just see what I can reach. So, like this kind of area right here?
Yeah, probably better like a little bit higher and away from the structure. Yeah. Oops. Yeah, that's the reason why I think.
Alright,
Or maybe is it possible to go on this side?
which side?
From coming from this side here, from the.
No, there's
Or
a big,
this one?
There's a big purple worm.
Okay, yeah, yeah, okay, okay. Or any other side, like here, or.
We could try it. Let me see how high I can get there.
Mm-hmm.
Follow me. What about that? I can get you that.
Yeah, I think that would be fine.
You want that? Okay.
So with the, we have a very, so we have the chamber number 5, right?
That's where you want it, 5?
Yes.
Okay.
And then just don't go in too much, but stay more on the surface.
Okay. Stuck?
Yeah, it's.
I'll try going the other way.
I going back.
You did go the other way?
Yeah, but I'm going both ways.
Stuck both ways. Okay. Put the port arm then.
See, try both ways and then make sure we're both going in the same direction. Just move over some. We got over some. Yeah.
Just do ice.
So we give it a shot. Can you tell which way it's going?
Right, yeah. Just don't do a little arm in there. You never let it out.
Yeah.
All right, come back.
That way, it's exactly the way. There it is.
Oops.
Backing up.
Wait, wait, wait. We are on almost on 5.
Okay.
Almost on 5.
All right, starts going. Next time, but don't go super fast, go kind of slow, but just don't stop, let it go. I'm backing up, okay, so go for it. Like I said, if it starts going, just keep going.
Well, I don't need to go much.
So this is a good time to reintroduce ourselves. This is live footage from ROV Subseafloor on the bottom of the Pacific Ocean. This is the Underworld of Hydrothermal Vents expedition. Just getting that device ready to go.
This away.
It's just a matter of how set.
Alright, watch. Alright, I'm ready for suction when you go. Just tell me when it's on.
Just interested in the small creatures. Like that, yeah. Around a little bit. Do you think that's staying here like that on the surface?
You need to touch a little bit because there are a lot of tiny little red dots everywhere. That's what Zabina is after. Well, that's, yeah, 5 is with the fine mesh, right?
Very fine mesh right now.
Okay, yes.
Yeah.
It's good. Move a little bit to the left. And just
I
keep.
see the red guys going in there.
Yeah.
Yeah. What is that?
Copper pots.
That's cool.
The pods, yeah. And there are
So
like hundreds of them, and Alessandro will be very happy.
Yeah, you see them all?
Yeah, so we can move around maybe a little bit
Yeah,
like
they're in there
that.
right now.
Yeah. Maybe we can do this for a couple of
There's a
minutes.
big group of them right here. Can you
That's
pan
great.
up a little? That's a huge group right in there.
Nice.
The team's trying to do suction sample.
Yeah. Yeah, okay, cool.
And we pulled up a view of
It's definitely
the
So
suction
like
sampler jar.
a few minutes, it's clogged.
No, it's
You
just
may
you
also
see
be
it
able to see
slowing down a
those
lot,
Yeah.
so it's getting
couple
plugged.
pods.
It's not surprising. I mean, there's
very
so
fine
much
mesh,
little
yeah.
fine stuff coming off.
It's not.
Even have a little shot. We'll see.
Yeah.
It works. Almost nothing.
But it's still getting the really fine particles. Just
I-
a lot more
Yeah,
slowly.
but I think now we're gonna switch to chamber number 1.
That was wait, what? That
Jar, sample jar, what?
So there's 8
Yeah,
suction
chamber
sampler
number
jars on Sebastian. The science team will preload those with different
Yes.
fine or coarse meshes,
So now we
depending
would
on what
move
they're
into
trying
a
to
little bit
sample.
more, right?
Okay, you ready? On one. And we're
So
trying to
they'll
do
Now we would move into a little bit more.
use
and, get maybe a little bit bigger stuff.
Jeez. And they track what's in what.
Yeah, go for it.
there it goes.
All right, here we go.
Yeah.
Nice.
Yeah.
I don't think you.
Beasts crawling around too. This is how you get ants.
So in this view, you can definitely see things flowing into that suction sampler jar.
Maybe go in a little bit deeper.
Alright, that's in there pretty good.
Is it still sucking, or is it?
No,
Yeah, it's
still
so
going.
dark.
It's still going.
Okay. Maybe over here. How far can you reach over here? Or?
To the left.
Yeah.
I think it's smelling there now.
A bunch of those red things. Yeah,
Oh, there's a bunch of these of the
Yes.
copper pots again.
Yeah, it's
But
definitely
it seems
slowing
like
down.
yeah.
Oh, just some of those worms too. I'm gonna get him next time. He's in here.
Alrighty, I think
I
it's
don't have enough suction right now though.
okay. So we can stop that now.
Okay.
And now we go to chamber number 7.
Course mesh, for sure.
Go really going in.
Go, yeah, there's some scale worms going around.
Yeah. I know where one of them is.
So now we're going into it.
You want me to get him?
Oh yeah.
Definitely. Coming up on
I see one there.
He's a good one. So,
Let me do that to
You talking about this white one over here? There was a purple one too.
Go for it.
Oh yeah, this one. Yeah.
Yeah. Should be a fairly easy catch. No. Don't care to look at. Unless that one clings very hard to its tube as possible. Ah, he's escaping.
no, I think he's in there by the number.
The turf flew.
Get him?
No, you didn't. I said crying out.
Let's try to stay away from the sculpture. Move a little further up.
So
Into
don't
it.
worry too much about going in, don't worry.
Yeah, go into it. Yeah.
Okay.
Albanella pompeiana.
Yeah.
Yeah. Orange banana. That's what I'm saying. Oh, see that guy right there?
Yeah.
He just went in there.
I didn't see that. I
That
trust you.
he was super, super, super long.
There. I saw one coming in.
Oh
there.
yeah. Oh yeah.
Oh, this one here.
That
Not
one.
necessarily want the albanella per se, but
Well, I can always use some.
Okay.
That's okay.
all right.
Oh yeah, there you go.
No orange vanilla, go ahead.
Almost.
Think he might have gone?
I don't know. The
Yeah, they
tube's still.
crawl away pretty fast. They don't trust you.
They're smart.
You might have to take his house as well.
I think you can help on that focus a little there.
Yeah. Sweet.
That
All right.
I can't do.
So it's still sucking, huh?
Yeah, we're still. We'll keep it running for a little bit on this one because there's so much in the tube.
You want to let it settle and see what you got, or keep going here?
There's a lot of stuff here, huh? That doesn't seem to be.
Oh, that's on the tube?
Oh, that's
Looks like
outside. Yeah, we can. If there's still suction, we can probably go in again.
Sure.
Oh, yeah.
Yeah.
Go back in?
Try to get as much as possible.
Okay. Any certain area?
I've converted you to the dark side.
Solid maybe. No.
Going down there in that.
This camera looks pretty.
What's that?
When you get in there, the wrist camera was pretty heavy.
Was it?
Yeah.
Alright, you look to the right a little bit, right? Get better. See anything else over here you want? Yeah, right there. Over that scale one back in here.
Oh, you gotta dig a little up.
Oh yeah.
Oh, man.
Worry.
We make efficient use of
Snowstorm
everything.
right now.
I want to see what's on the other side of this. I guess it'd just be a rock, wouldn't it?
Yeah, chimney, basically. So they colonized the outside of the chimneys.
Oh, there's a big old fella there. He's
Oh yeah.
he's chunky.
That's close to the art.
Oh, yeah. Oh, I can't see it. I can't see
Oh. It's coming, sort of. Resisting.
Yeah, I think so.
Wiggling out of its tube right now.
Enough.
Good.
Yeah.
Thank you
Okay.
very much, guys.
That's awesome. Let that run for a little bit.
Yeah, sure, it comes through there.
Yeah. Wait till.
There it is. There's one.
Is that coming?
Yeah, he made the
Oh yeah,
Should be more coming.
there are quite a few in
There's a
there.
lot of suspended matter.
So are these guys gonna all be mad and like fight each other in there now?
Well, in pissing themselves because right now we put them at 2 degrees Celsius when they are used to 20.
Alright, so we still have that metal hook to hang this on, huh?
Right there, where the handle goes in. You said a white. I know that. That's part of the holster. I can't see it. Want to pour it back in?
If we-
I think
if
we'll
it's
see,
possible
sure.
to out there, that's in there, but
It's a good shot. Right?
yeah, exactly.
Yeah.
At least there's a chance.
Oh, totally.
Oh, okay.
So I guess we have to go to crab spa now.
Pour chocolate, you know where it is.
Just a bump, one bump. One bump out. Oh, a little higher.
Yeah, you have that much time for
Right. One bump.
Oh.
No, you know, I'm just gonna drop it. It wasn't that hard to get anyway.
No.
It goes there great. Almost. All right. Whoop.
Where's it?
Oh, if you get too near the edges, it will.
Okay.
Like if you look too far down, it'll drift up. If you look too far up, it'll drift down. That's all I've noticed.
So now we are moving to a grab spa, and actually the location that's named Stefan Grab Spa. So that's exactly the place where we want to be.
Is that named after you?
Yeah, that's Stefan.
Yeah.
Named after you?
No. I mean, this is the name is Grab Spa, which has.
Well, you guys might be famous, and I don't know.
Well, Stefan
So-
did name Crab Spa.
Yeah.
All right, so you're famous.
Yeah. No, I mean, yeah. So Grab Spa is a kind of an interesting place. This site that we're going to now was discovered in 2007, 1 year after a volcanic eruption that occurred at this site, which we're currently studying, which wiped out basically all life, or at least all animal life. Microbes were still present, and they were recolonizing these vents very quickly. And so when we came across Grab Spa, there was a lot of flocculent material being released from the subseafloor environment. There was white staining biofilm on the rocks surrounding the outflow. And what we also observed were crabs actually that were hanging out there and going in and out and feeding on the flocculent material that was coming out. And so because the surrounding seawater is like 2 degrees centigrade and Crab Spa, the fluid is like 25, we thought it was kind of fitting to name that site Crab Spa. where the crabs are hanging out and enjoying themselves. And we have been sampling this site frequently since then, since 2007, as much as we could. And so the last time it was sampled was in 21. And so we have a relatively good understanding of the changes that occurred at the site, both at the surface as well as in the subseafloor environment.
Stefan, would you mind introducing yourself and where you're coming from and
oh yeah, of course, sure, yeah, thank you, yeah. My name is Stefan Sievert. I'm a scientist from Woodsole Oceanographic Institution, and my role on this cruise actually is to characterize the microbial communities, the diversity and function, and with a specific focus on the subseafloor environment. And a peculiar feature of crab spa is also that it has a quite defined orifice where the fluid is actually coming out. So we could repeatedly go back to the site and get the same type of fluids whenever we were here. So that makes it also very reproducible, and for if you want to look at any changes that occur over time. So it's actually situated on top of a spire for 504. Yeah, I think we are now at the base of the structure. I think if we move up, we're probably gonna. Yes, 2,504.
Right, so we were, I was going down because we were shallow. I see.
I didn't know what depth it was, so it's below us now.
No, yeah, now we're at 2,507, so it's 2,504.
I think I don't know how fast that updates. I'm looking at the depth up here.
Yeah, but we are, yeah, so this is actually. So I think it's actually on the, let's see. Yeah, this is actually it, I think, yeah. Yeah.
So what are we doing at the substation?
So we are going to take a water sample, but also potentially riftia and mussels as well. So I'm just trying to confirm that we are at the correct site.
We're going to spin around this thing.
Yeah.
So when we first got here, I remember we had some trouble finding it. And
Yeah, I think we
found-
were first at this site on over here, but I think this, yeah.
We got one called Crab Spa and one called Crab Spa Falcor 2. So we can go check that other one out too if you want. It's just right there.
Yeah.
You want to check it out?
You see what
Yeah,
you're
if you
talking
can,
about?
yeah, I know. No, not this one. I think it's, yeah, it could be over there, but I think that this is, yeah, let's see.
Okay. You happy? We're happy.
We can maybe move around quickly. Then actually there should be Tika Spire very close to it, right around here. There should oops, sorry. There should be weights actually, some weight stacks down here. I think that's the that's web spar. I'm pretty sure. Yes. Yeah, there weights. Yeah, right, exactly. So yeah, we can go back here. That's it. That's it here.
So at the bottom of your screen, you've got some weights, and that's what Stefan is using to identify this as crab spa.
We should get a sign.
It'd be easier, right?
The upkeep around this field is terrible.
Yeah.
So yeah, we need to somehow position ourselves in front of it, and then
Where exactly do you want the water sample?
within here. So we just need to find the warmest spot, which should be around like 25 degrees.
Sure.
But I think we can probably first maybe sample the rift here. They seem to be, I think they seem to be in a good size, potentially for Chessie.
Preferably not free flying. If you can find a way, maybe you could bump your leg bumper against the muscles or something. Or I guess if we have to, we can free fly it, but
Yeah, no, I
it would be all over the place, knocking it over.
Yeah, so for taking the water sample, it would be really important to have a stable positioning so that we are at the right spot.
I think you could brace there, you'll just be pretty high where you are. It's doable.
It's a little crazy with that suction hose and that. I can't believe we know.
Yeah. You should have seen it before.
You should have said something.
You have to porch out to get the probe.
Yeah.
So if you guys, if you think it'd be easier to just do that while you're free flying, or sometimes it's a little tight if you get in here and have some stuff. That's probably
I'm fine if you can do it.
Yeah, we will need the temperature probe.
Amazing.
It's the best porch I've ever seen. Everybody say it.
I'm not saying that. Everybody else.
Man, somebody really put a lot of thought into where that thing is.
So, Stefan, this is a temperature something?
Yeah, no, we are proceeding with measuring the temperature.
Yeah, just kind of there.
Oh, got it. Right.
That kind of right there.
Like over
You
here.
can see real well on the 4K.
Yeah.
Just stay ready to pull back if it does push through.
Can you take a
Real
frame grab?
forward, you just down on that piece. I would put it on top of it, just pressing down. Well, you might be able to do it.
On the spot or the one above it?
I think you would.
Are you good?
Just watch his cable. Yeah, we're already pretty low. See if you can put that on the top of it. It'll get you a little higher.
I don't know if there's anywhere, if this doesn't work, if there's anywhere we can do the left roll, then you know, in the room.
Yeah, the hell you change it.
Yeah. Yeah.
Right up on here.
Well, look down at
tiny bit for
Yeah,
you.
okay. Not right now, but soon, yeah. Thank you.
Give it a whirl.
Just a second, let me see if I pull the picture. So,
Zoom.
Yeah.
Light.
Probably more like further up, I think. I think further up, like and actually go in.
Further up, okay.
Yeah.
Do you want any additional text here?
Oh no.
is that your?
Tells me. Give me some focus.
Autofocus.
Stefan, do you want me to write any additional text here?
No, I mean I think yeah we are trying to find the warmest spot
17.
right now, and then yeah.
50.
Can you move in a little bit more?
No. We're going to be able to not really be able to see though.
Yeah.
19.9, 20.
Yeah, just leave it for here for now and just see where it goes. Yeah. That's a pretty good spot, I think, but we can maybe check out like a little further up.
Okay, you want me to come out of here?
Yeah, just remember this, we could go back. I think this is a good spot, but just want to try somewhere else.
Those eggs.
Where?
Right where I'm pointing here. This,
I think these are Paravinella, so they're close relatives to Alvinella. And they just make a mucus and they tend to accumulate little pieces in there.
almost looks like pebbles or something, you know?
The orange ones, right? The bits? Orange bits?
In between the orange bits.
In between the orange bits. Oh, sorry, these are limpets.
Yeah,
or
it's
little
all
bits
over.
they accumulated in their tubes.
Yeah.
Nine, 10.
Yeah.
Should I take all this temperature measurement?
No, we're just trying to be looking at different areas. To maybe you can actually go here.
This?
Yeah. Right there.
Like from the particles that get stuck in the mucus from
So it's one possibility, the other one is actually bacterial or current boundaries of the surface cheaper than regulations.
That freeze point.
Yeah, it seems, I think the freeze drop is the pitch. And
Oh,
pitch
it's
down.
actually-
right?
That's perfect.
Yeah, it seems like the other place was somewhat warmer, right? So I think.
Go back?
Yeah, I think so. I think it was down here, right? In this area.
I think it was that one, but you
Yeah.
want me to try this one first?
Yeah, we can try there.
And then we will have tried them all.
Yeah. That could be a nice spot actually.
Way.
Yeah, when we do the water sampling, it would really be important to remain stable.
Yeah, I think when we get to a good temperature reading, I'm gonna freeze the arm, and then we'll do the water sample without moving the arm. Would be the best way
Yeah,
to do that.
because we need to stay there like for 30 minutes.
Yeah. But if I freeze the arm, should be fine. But we just came off because.
Yeah, sure, yeah.
Punch the rocket.
Right, okay.
The water sampling is with the blue bag, right?
Yes.
So that will be the other arm and the other won't.
Oh yeah, it's right, yeah, so yeah, thanks, yeah. Oh,
Sorry.
Yes, thank you.
Well, that's a
Yeah,
good
we're not
news.
taking a water sample with this one because we already took one.
We
Thanks,
can
Stefan,
still,
yeah.
We can still freeze it.
Would you mind making it so I can see the starboard ram with the second? Perfect, thank you.
Yeah, but I think the last spot where we just were looked like it might actually be quite good. I think that was this here, right?
Yeah.
And but I also think these riftias look a good size for chassis. So I think I suggest we take them actually after taking the water sample.
That was simple, so I want to go too much over like 25 trims.
What's that?
I'm trying to keep the trims at like 30.
Yeah, you will push on these right over.
Yeah, I think that's, might have to try with it.
Are you happy then?
Yeah.
Oh, wow, wait a minute. Wait a minute. Can I look at something real quick?
Do it.
Hold up. Look at that sea spider.
Oh, no good. Yeah. Whoa. Pretty big one.
You don't see those very often. That guy's
Do a
like
crimp
face
wrap.
size. Perfect
Sea
face
spider.
size. Look at him, just waiting to jump on you while you sleep.
It's better from nightmares there.
Wow, I've never seen one this close.
Nice. Good catch.
That's so cool. It doesn't even look like a real creature, does it?
No. Doesn't look like there's enough mass to accomplish.
Isn't it their guts are in their legs or something, and that's why their bodies are so small?
I don't know.
There's the pair of green leg-like lips on which they attach some of the eggs.
Alright,
Anyone else care?
sorry.
Oh, okay.
Get distracted.
No, that's nice. I mean, yeah, thanks for catching that.
Alright.
So I think it was Where was it? This one or this one? I think it was this one here.
Oh, I know where it was. I was going to go into that one.
Yeah,
but
yes.
Originally, we were in that one.
Yeah, no, but I think this one we tried just when it became
It was
unstable.
looking really good right until Alex
Yeah.
totally blew it, you know?
Yeah. It's,
I'm totally harassing, by the way. I punched myself with the
that's, no, that's okay. I like driving off. That's okay.
Not California, but else.
Yeah,
We'll stay on.
that looks really promising.
That's a good question.
I'm froze.
Okay, when you froze.
20, 20.8, 20.9, 21.
Yeah. Okay. Can we just go one down?
One down. Okay, coming out. I'm freezing. Straight
We can
back.
remember this one, I think. So, but it was like 20 degrees.
All right, and so you want to go back to the original or somewhere else?
Yeah, just the original.
Original?
Yeah. So what do you think? I mean, is this so you know the one or the other with the blue bag? No.
All right, so we're going to put this away then? Put the HC
Yeah, I think. I mean, either we try this one here, which has the highest temperature, or then if not, then we try this one here.
Okay.
Yeah.
You want to push out a little bit?
Yeah.
Is Stihi there?
He just walked out.
Oh, okay.
Precarious about.
Maybe you can
What do you
come
need to
back.
know?
I mean, he's usually here when we do the pump.
Should I go for me?
Well, yeah, because I don't know if he's actually watching what's up.
Yeah.
I think about.
Clarissa, would you mind
I'll-
getting Stihi?
Okay.
Thank you.
Coming up.
That was easy.
Oh, there it is.
You heard
Okay.
your name, right?
Yeah.
While you were watching.
So we're about to start.
He's watching us. Be aware.
Who?
So, it's watching the bag.
Yeah, you're
That's
not
not us,
yet.
it's the
You
bag.
have to first put in the wand.
Can you come out anymore? Or
Yeah.
Oh yeah. Just want to keep it inside of the set. Do you care if it goes outside on the set?
let me just give it a try.
So 77, right? The water bag.
So after using the high temp probe to locate the
What's that?
warmest vent flow in this site,
Did I miss something?
pilots are going to stow that probe.
77.
And they're going to grab a different sampling device. And they're going to fill the bag that I just put up on the screen that holds about 55 liters of
Oh, yeah,
water.
you're right. Okay, thank you.
And there is a pump that the pilots operate via the manipulators. And so they get the wand for that pump in the proper spot, turn the pump on, and we'll wait a little bit while that bag fills with water to be sampled. Bring it up to the scientists here on RV Falcor 2.
And that is this fellow here, huh? What's that?
Yeah.
Don't grab it, Tina?
Yeah, grab it by the.
Wait, where to grab?
Yeah.
speak up a little bit?
I was gonna say, the hose is kind of.
Let me do something, or I'm okay?
It'll take that. Okay. Yeah, it was a little worse.
I don't know if I can reach over there or not, looking at the reposition, if you try and pull on it a little bit, I can pull on this, right?
I'm not sure how long it is.
Yeah, it's different types.
If you need more hose, all you gotta do is porch out. Or just porch out. Sorry. It's okay. I think I'm gonna pass it to the start.
Will you get the whole bath from here, Stefan?
Yes.
The whole bath?
Yes. Yeah. Was it this one here, right? Or? No. Yes.
Okay.
I think, where was it?
That
I
was
think.
where it was, right down the hill.
Yeah.
Wait, how are we gonna turn the pump on?
Maybe lift it. Oh, we have to turn the pump on with the starboard arm, huh? Oh, man. God bless America.
Oh.
Alright, well,
You need this arm for turning on the pump?
Yeah. Yes.
Oh.
I guess we're just gonna have to move, because we're not gonna be able to do that. Alright. Sorry, this is an awkward spot, fellas. So I'll pass this back to the port.
Okay.
You reposition, put your bumper near that, and then we'll.
At the port ramp?
Yeah. We're gonna have to be on the port ramp. So good for that too.
I could have not said anything there.
So this illustrates the many considerations the pilots have to take into account when doing things. So the best way to get that sampling wand into the vent was with the starboard manipulator. But in order to turn the pump on, you have to use the starboard manipulator. So you're gonna shift the sub around, make sure the right arm is doing the right thing. So it's difficult in a situation like this where they're bouncing on one ram on a fragile vent and trying to determine the best way to get at the sample site. And for the question that just popped up on YouTube, So the instrument's in the manipulator now. So it's a sampling device, so it's a pump. So attached to that is a pump and a bag that the team will fill with water to bring back to the surface. And we'll throw that up on the screen right now. So the pilots will maneuver that wand into the site that Stefan has identified as where he wants to sample from. Turn on a pump, fill that bag full of seawater to sample, or vent fluid, so they can bring it up to the surface and give it to the scientists to run some experiments on.
Just the bottom left.
Try and just put your port rail against something. Just remember, it doesn't have to be just down. It can be forward too. You know, like maybe you could even put it against those muscles if they're hard enough. I don't know.
Probably go too low.
Tough one. You just have to try different things until something sticks. I think. You know?
That ledge or below it, it looks secure. I think we can reach. That's all.
Just try to get where we want to see now.
The end of it there.
You know what we need too, if we need to, we can torch out and run the Portuguese system.
And they'll try to get that balanced somewhere where they can still get to the sample site. Get the thrusters tuned in to keep it not moving, lock those in place, but as most things we're seeing down here,
Yeah, let's just try that with the
it's
Portuguese.
not as easy as it looks.
Alright, you should be able to just torch out.
Just be where you can walk that against you.
So to give themselves a little bit more surface area on the sub to work with, use the porch for this.
So also what you see here in the water is a lot of particles that are coming out from this vent site, and most of it is actually, there's some biomass of course associated with that, but a lot of it is also sulfur in elemental form, elemental sulfur, and that's being produced by microorganisms living in the subseafloor environment. So they live below the ocean crust, where hydrothermal fluid and seawater mix and do chemosynthesis by oxidizing sulfide with oxygen and form elemental sulfur. And so they live in the cracks and crevices in the ocean crust, in many cases as biofilms, and some of that is then being sloughed off by the fluid flow, and then this is being ejected here at this location, for example.
It's using a little bit of lens.
So we can see this basically as a window into the subseafloor environment, and we take the sample here to get an idea what the subseafloor biosphere is like, what type of organisms live there, you know, what the functions are, and how active they are. And once we are positioned and take the water sample, it will take like half an hour to actually get that done. There will also be an opportunity by Clarissa Karthäuser to talk a little bit about these particles and the work that she's actually doing in characterizing these particles, which is very novel to do this here at the vents. That usually is being done in the upper water column to look at marine snow, but no one has really looked at the particles down here, and so she's doing experiments, but she will talk more about this herself soon.
We're looking rock there,
I think you're up to the right.
huh? It's
Was
just-
this the location?
It's just a different angle. Yeah, 'cause-
Yeah, okay.
Alright, froze. Yeah, that's where we were, where we got the highest.
Okay. I'm not totally sure, but
It is Stefan. No,
yeah.
It is. On a wider angle. And you see
Yeah.
there's like little beads with the power line just above it.
Yeah, okay.
I just remember because the limbits were right above it.
Yeah, okay. Is it as far in you can go?
Yeah.
Yeah, I don't want to push because I'm not
Yeah,
soft.
okay. Are you stable right now? Like the sub stable and everything?
I'm not stable, but I think the sub is pretty stable.
Okay. So okay.
Don't push it.
Okay, great. Though then maybe let's turn on the pump, and then we'll pump for like half an hour.
So first we put out X.
What, you want to pump it out?
Not yet on, just
out. That's it.
What's that?
We're going to go from bag to EXP first.
Are you racked in all the way?
I am wrapped in as far as I go.
This is going to be a little weird to you.
Yeah, you have a lot more room than it was. So, HT2.
I just don't want to hit my other minute, you know?
Yeah,
So,
look at it.
Don't we have to go to bag because it's going into the bag?
first out, 2 minutes.
Okay. Oh, all right. Okay. You know your stuff. Okay. Yeah. Okay.
No problem.
I'll be quiet.
No problem.
Nothing broke.
everybody alive?
Looks like it.
It's great.
Just a little on the cooler, but it's not as bad as yesterday's.
Sound okay?
Yeah. It's gonna happen.
Alright, go EXP?
Yes,
Yes.
yes.
I think what we did before was we went to
Yes,
EXP.
I mean that it's okay.
Yeah.
Stop. Yeah, good.
That's fine.
I'll stop.
Yes.
Then go up on.
Now go home?
Oh, yes.
And then for 2 minutes.
Yes.
And then go back. Then we go back to that.
Yes. It's okay.
Okay.
I mean, it's okay. We test.
So now 2 minutes.
Yeah, no, it's we test out and okay.
If for some reason we come off of here, Alex, just make sure you go straight back. 'Cause
Yep.
I can't move the arms, you know, I won't be able to move both fast enough.
Yep. Understood.
Remove this.
All right.
Now it's back. Not power.
Just
power.
barely below. Yes.
No, we don't need that.
Wasn't doing that?
That will outgas a little bit.
I think that was just focused on it.
It's filling. I see the bag inflating.
Great. Okay. Cool.
I can still get this more towards the bag. This is see what we get. We can do this.
I think that's like group.
That's it.
Nice. Thank you.
It will be on this maybe for
35 minutes?
Just one more?
30 minutes. Yeah.
30.
Oh, we'll see with the bag. Yeah, but I think about 30 minutes should be okay.
Yeah, it turns on like the first quarter, like cafe.
Okay, so now we are pumping fluid, basically coming from the subseafloor. So we have a tap into the subseafloor biosphere, and we're sampling the water and any microorganisms or other type of organisms that are associated with it into the bag, and then we'll take that water and do different types of analysis with it back in the lab. Or even back home. So we'll preserve some of it for later extraction of DNA and RNA, for example, to look at the diversity and the functions of the microorganisms that are in there. But now I would like to turn it over to Clarissa, and she will talk a little bit more about the type of work that she's doing to characterize these marine particles here. Clarissa?
Hi everyone, I'm Clarissa. I'm a postdoctoral scholar at Woods Hole Oceanographic Institution. And whenever you look at the camera, you see all those little snowflakes, and we actually call them marine snow. And I study what happens on the marine snowflakes and stick together and sink into deeper water layers. Marine snowflakes can form, and they transport biological material through the water column. But since at the seafloor we also have fresh biological material being produced by the organisms that use chemistry instead of light as the energy source, we can also have lots of fresh snowflakes there. And each snowflake is very tasty, of course, for bacteria. They like to feed on them. And I'm trying to find out what bacteria do with the marine snow. So what kind of bacteria are on there? Do they breathe? Do they do other chemical conversions? And what does that mean for the overall system?
Saludos, hola, buenos días o buenas tardes o buenas noches de donde sea que nos estén escuchando. En este momento están viendo el live stream de una expedición a bordo del Falcon 2, gracias a la transmisión que tenemos a través del sumergible, el ROV Subseafloor del Schmidt Ocean Institute. Yo soy Iván Rodríguez Ramírez de la Universidad de Costa Rica y les voy a estar acompañando en la siguiente hora con narraciones en español sobre lo que estamos viendo. En este momento estamos tomando muestras de agua y estas muestras de agua van a ser distribuidas entre los diferentes grupos del equipo científico para diferentes análisis. Una parte es dedicada al estudio de los virus, otra parte es dedicada al estudio de las bacterias y otra a la potencial, a la potencial diversidad, no tan potencial porque de fijo hay mucha diversidad en estos ecosistemas, de meiofauna. El día de hoy en el micrófono principal nos acompaña el doctor Stefan Schwiebert y vamos a estar hablando con él un poco de todo lo que se está haciendo el día de hoy. Hi everyone, for the non-Spanish speaking folks, we are entering right now the Spanish section of the dive, but as always you can send your questions or your messages in the chat in English or in Spanish or in whichever language you feel more comfortable with. Vemos que tenemos en el chat una pregunta para Clarisa. Clarisa se acaba de ir de la sala de comandos, pero apenas vuelva le pasamos la pregunta.
No, that's fine.
So all the round things, they're not limpets, they're little snails, coiled snails, Cyberthemia, Microbiological, and they live in very specific conditions so far. Ian, who's working on gastropods, has had only a few of them. So here they're everywhere.
So we could go back here.
Yeah,
we don't have a slope left.
Yeah, not today, but maybe another day.
Yeah.
If you need more Cyberthemia, that's the place to be.
Yeah.
What size mesh would you need for those?
Of course.
We have one course though.
But it needs to be used on key vents, right?
Yes.
Thank you, though.
Thank you. Yeah.
Good thinking.
One course and two.
I mean, we could check with Sabine.
That will be fun of her.
Yeah.
I mean, we'll be in that.
We can,
area,
yeah,
okay?
exactly.
So even
Exactly.
when we are collecting the mesh boxes, if we have time, that's an easy place to come to.
So is this par vanilla or vanilla?
Para vanilla.
Okay.
I'll make sure Ian knows that.
Thank you, Stefan.
Keep doing close-ups.
Yeah, no, I agree. Yeah. Yes.
Do you want to explain what we're doing right now?
Yeah, sure. Yes. So my name is Stefan Sievert. I'm in the USA. I'm originally from Germany, but I went there as a postdoc back in 2000 and had the opportunity to stay there as faculty. And so I've been living there for the last 20 plus years. I've had the privilege to actually work at deep sea hydrothermal vents. It's always really great to come back here. Been working at this site for the last almost like 20 years since my postdoc. And this place here is quite special to me because we found this place called Grab Spa back in 2007 after a volcanic eruption that occurred at this site. And at that time there was basically no animals after this eruption, but just bacterial mats, like biofilm on the rocks, and a lot of floccular material coming out that we still observe today, as you can see here. So that's basically biomass that grows down in the subseafloor environment and then is being released at this vent here. So we can use it basically as a entry point into the subseafloor environment. And over the years we've been able to come back here repeatedly. We know the location, we know pretty well where the water comes out at the highest temperature, and so we can get reproducible samples, which is really important if you want to look at how things might change over time. And at the surface, of course, this site has changed a lot. So since the beginning, then different types of tube worms colonized. So first, usually it's Tephnia that colonizes, a tube worm that's more adapted to higher temperatures and higher concentrations of sulfide, and then that gets replaced by Riftia, and eventually mussels move in. So what we see here now is kind of a climax community of Riftia and mussels, and also you see a lot of smaller organisms. So there are snails, there are limpets. So it's a very diverse kind of system here, and there's an immense productivity that happens above the seafloor, as you see here, but a lot of it also happens below the seafloor. And so that's what we are interested in. And so what you see here is now the wand stuck in the vent and sampling this vent fluid that's coming out from below. And that contains not only the water, of course, but also any kind of organisms that are associated with this and that grow in the subseafloor environment. So we sample this into the bag that you see maybe inflating. Sometimes the camera is on that, and we will collect about like 50 plus liters, and then we'll bring this to the surface, measure chemistry, and also collect the water for different types of experiments and also analysis. One group here is looking at viruses, for example, and productivity of viruses, how soon bacteria release viruses, for example, so bacteriophages, and we are particularly interested in the microorganisms, bacteria, that are living in the subseafloor environment, and to look at their diversity and their function. And not on this cruise here, we don't have the means for that, but in past cruises we also have done incubations to look at the activity of these microbes using special devices that can maintain the pressure of the water, and then we come back to the surface and can perform experiments under simulated seafloor conditions.
Bueno, muchas gracias a Stefan. Stefan nos estaba contando primero un poco sobre él. Nos decía que, bueno, él es un investigador alemán, pero que ahorita está trabajando en un instituto en Estados Unidos, y que este sitio para él es muy importante ya que se viene estudiando desde el año 2007. En el año 2007 hubo una erupción volcánica en esta zona, y entonces las comunidades que estamos viendo en este momento en realidad son bastante jóvenes, son bastante nuevas, y fue una manera muy bonita, dice él, estar estudiándolo todo este tiempo, porque entonces se puede ver todo el proceso de colonización del sitio y de sucesión de diferentes especies que se ven acá en esta zona. En este momento estamos tomando muestras en estas ventilas hidrotermales, y Stefan nos contaba que estas ventilas son como una especie de puerta para las comunidades que hay en el subsuelo. Stefan está enfocado en la parte de microbiología, él es un microbiólogo marino, entonces su área de expertise es justamente los organismos que viven en la corteza terrestre en estas zonas. Él nos contaba que lo que han visto de sucesión es que primero estos sitios son colonizados, bueno, pues por bacterias, luego siguen unos gusanos poliquetos del género Tephnia, luego estos gusanos son, digamos que desplazados por el género Riftia, que es el que estamos viendo ahorita en pantalla, el que tiene estas plumas rojas tan grandes. Luego de los Riftia vienen estas comunidades de almejas que vemos en pantalla también, y después de eso ya viene todo el resto de animales. Entonces está muy interesante porque la comunidad que vemos en este momento es como una comunidad clímax básicamente de estos ecosistemas, como el último paso en todo este proceso de colonización de un ecosistema de una ventila hidrotermal. Luego también Stefan nos contaba un poco de para qué se utilizan las muestras de agua que estamos tomando en este momento. Y bueno, justamente también lo que mencionábamos hace unos minutos, ¿verdad? De que una parte de la muestra va a el estudio de virus, de ver bacteriófagos que pueden estar presentes en este agua. Otra parte va para un estudio microbiológico enfocado en las bacterias, en la diversidad de las bacterias. Y también nos contaba algo muy interesante, que es que en cruceros pasados se podían hacer incubaciones de estas bacterias para ver cuáles son sus características específicas de manera in situ. En este momento, en esta expedición, no estamos haciendo esto, pero es muy interesante saber parte de toda la investigación que se ha hecho en la zona. Ahora vamos a traer de nuevo a Clarisa, porque hay una pregunta para ella en el chat, que la pregunta es justamente sobre cuál es el porcentaje de las bacterias que Clarisa estudia, ¿verdad? De las bacterias que están presentes en las partículas, cuál es el porcentaje que es conocido, y si ella aísla esas colonias de bacterias. Entonces vamos a ver cuál es la respuesta a esta pregunta.
Hi, so I received the question, which percentage of the bacteria in the marine snow are known to me? And do we bother isolating them, or are we looking at colonies in general? And so I've actually never worked with deep sea marine snow before. And also I don't know anyone who has looked at deep sea marine snow, individual snowflakes. So this is going to be a very big surprise. I intend to get DNA out of the marine snow, and I will use a technique called barcoding. So like items in a shop, you can use DNA sequences to recognize different bacteria based on sequences that only the certain species has. And I plan to do that to know which bacteria are present in the marine snow. But especially in very remote places, I expect to be a lot of bacteria that nobody ever had a closer look at or isolated. So I expect there to be a lot of bacteria that we do not know. And for that, I can use a technique called metagenomics to at least learn a bit about what they can do. And with metagenomics, we get all the DNA that is in the marine snowflake out. And then we can take it and check which genes are there. And genes basically are the plan to build components in a cell, like enzymes. And if you know which gene is responsible for which component, you can know, for example, if there is an oxidase, that the organism can maybe utilize oxygen. Or if there is a sulfur oxidase, it can do sulfur oxidation. And so I can use the metagenomes to learn a bit about what the bacteria can do, even if I do not know the species yet. So I hope that answers your question. Thank you.
Gracias a Clarisa por la respuesta tan genial que nos acaba de dar. Bueno, Clarisa nos estaba contando justamente que el proyecto que ella está haciendo, la investigación que está haciendo es sumamente novedosa. Entonces, en realidad no se conoce mucho sobre las comunidades de bacterias que están presentes en la nieve marina, ¿verdad? Que son todas estas partículas que vemos ahorita en pantalla, que es con las que Clarisa trabaja. Entonces, ella está utilizando una técnica que se llama barcoding, que es básicamente buscar los genes que son específicos para cada especie, para poder identificar especies nuevas de bacterias. Y ella espera que hayan muchísimas especies nuevas, ya que justamente casi nadie trabaja con las comunidades bacterianas que crecen en estas partículas. Después nos estaba contando que ella va a utilizar una metodología que se llama metagenómica, que es básicamente extraer todo el ADN que esté presente en esta partícula y ver todos los diferentes genes que hay. Nos estaba explicando también de manera muy clara que los genes son como esta información de dónde salen las capacidades o las características que tiene cada bacteria o cada organismo. En los genes está escrita la información base para poder llegar y tener cierta habilidad, ¿no? Entonces ella, al saber cuáles habilidades están relacionadas a cuáles genes, puede tener una idea de qué es lo que hacen o de qué son capaces de hacer las bacterias que están presentes en las partículas, a pesar de no saber cuáles son las especies específicas que viven en ellas.
So something that might be of interest is that, as I mentioned before, we did in situ incubations. So we collected seawater, vent fluids directly from this place here, brought it up to the surface, and incubated under keeping the pressure, and then incubated under different conditions, adding different substrates. And this way, trying to get at the question like how active are these microorganisms and how much biomass do they actually produce in supporting this ecosystem. That hasn't been known at this point, especially for the subseafloor environment. And what we found is that these organisms are really active. So they have a lot of energy available and they grow quite fast. And also to our surprise was a little bit that when we compare the productivity of the microorganisms below the seafloor with that what happens above the seafloor, and you can see how much actually is being produced here, it's almost the same. So as much as production you have above the seafloor, which is quite impressive, it's kind of rivals the production in the tropical rainforest, as much you actually have below the seafloor by microorganisms doing chemosynthesis. So overall, you can see these deep sea vents as kind of oases in the deep ocean, because you just move a few tens of meters even away, and there's like just bare basalt with almost no signs of life. And so here you have an additional energy source coming from the inner earth that's supporting all this activity here. Similar as you might see in a desert oasis in a desert, for example, where water is missing, and then if you supply water, everything blooms and grows.
Esteban nos estaba contando que bueno, justamente en las investigaciones pasadas en las que se han hecho incubaciones sobre los fluidos que salen de las ventilas hidrotermales, han podido ver que los organismos que viven debajo de la corteza terrestre son igual de productivos que los organismos que viven sobre estas. Y que algo que me pareció muy bonito de lo que acaba de decir Stefan es que estos lugares, zonas de ventilas hidrotermales se pueden ver paralelos a un oasis, porque la producción que tienen se puede comparar con la producción que hay, por ejemplo, en un bosque tropical, que son energéticamente bastante activos. Y nos decía que justamente si uno se aleja un poco de estos sitios, de estos ecosistemas, a los lugares en donde solo hay basaltos, se ve que hay una disminución bastante drástica de la cantidad de actividad animal, microbiológica y de movimientos energéticos que existen a través de redes tróficas. Entonces, estos sitios en el fondo marino son paralelos a ver un oasis en el desierto donde surge agua y crecen un montón de cosas, ¿verdad? Crecen árboles, llegan animales, aves, etc. Acá es lo mismo. Acá el flujo de energía, los nutrientes que salen de las ventilas hidrotermales son como esa fuente de energía, de nutrientes necesaria para que surja la vida, lo cual es algo muy hermoso. Si tienen preguntas específicas sobre la expedición, sobre cómo se estudian estos microorganismos, por favor mándenlas al chat para que Estefan nos pueda responder.
Yeah, and there's also a story behind the name. So why is it actually called Crab Spa? And so when we discovered the site, as I mentioned before, there was no riftia and no mussels, for example. But the only thing that was hanging out there were crabs, and so they were around Crab Spa and feeding on the floccular material that came out, and were going in and out of the warm water. And as you might know, the surrounding seawater is very cold, only like 2 degrees centigrade. And Crab Spa at that time was around 30 degrees centigrade, and now it's like 23, what we just measured today. So it's significantly warmer than the surrounding, and so we thought it was fitting, to name it the way we did.
In what year was that, Stefan? In what year was that?
Oh, yeah, so that was in 2007,
okay.
1 year after the volcanic eruption. Yeah.
Cool. Estefan nos estaba contando una historia sobre el nombre de este sitio. La locación en la que estamos en este momento se llama Crab Spa. En español sería como un spa para cangrejos. Y es justamente porque cuando vinieron acá en el año 2007, que fue justo después de la erupción volcánica que hubo en la zona, vieron que en esta ventila hidrotermal no había toda la diversidad que vemos en este momento, sino que lo que había, la especie que colonizaba el sitio, eran justamente cangrejos, que son la misma especie que a veces aparecen por acá, que son estos cangrejos blancos. Y entonces decía que todos estos cangrejos llegaban y entraban y salían del flujo de agua que estaba saliendo de la ventila hidrotermal, que recordando un poquito y también viendo, si han estado viendo las mediciones de temperatura, el agua que sale de estas ventilas hidrotermales es sumamente caliente en comparación al agua que está alrededor del sitio. En este momento en pantalla podemos ver en la esquina inferior izquierda que la temperatura en este momento en el agua alrededor de la ventila es de casi 2 grados centígrados. Pero si se hacen un poquito para atrás en el live, van a poder ver que las mediciones de agua que están saliendo ahorita son de casi 20 grados centígrados. Entonces es una diferencia bastante grande. Y bueno, por eso le estaban diciendo que es el spa de los cangrejos, porque los cangrejos estaban acá nadando en un agua bastante caliente. Stefan?
Yes.
There's another question in the chat asking what's happening right now. What are we doing in this exact moment?
Yeah, good question. So in this exact moment, yeah, I can maybe move to actually show that. We see here a hose that's connected to the bag in the back of Sebastian. And you can see that it has a wand attached to it that actually is inserted into the vent. And so we are currently sampling water that's coming out from the subseafloor environment into the bag. And that water contains microorganisms and other types of organisms that live in the subseafloor environment and that are being ejected here at this site. And so we use this as a way to get access to the subseafloor environment to see what type of organisms live down there and analyze them with different methods and do experiments with them as well to see the diversity but as well their function.
Estefan nos está respondiendo la pregunta de qué es lo que estamos haciendo en este momento. Entonces, para las personas que apenas se están conectando, muchos saludos. Acá les habla Ivonne Rodríguez Ramírez de la Universidad de Costa Rica y estamos con el doctor Stefan Schwiebert. Y él nos estaba contando justamente que en este momento estamos muestreando agua que está saliendo de la ventila hidrotermal. Acá se puede ver en pantalla que está como una especie de varita que es el apéndice que está conectado a la bolsa. Nosotros le decimos la bolsa azul, a pesar de que en el video acá en pantalla no se ve el color, pero es una bolsa azul muy grande que está conectada a esta tubería que está insertada dentro de la ventila. Entonces la bolsa en este momento se está llenando, dura como media hora para poder llenarse completamente. Y luego esta bolsa viene con el ROV a la superficie y el agua se reparte en los diferentes grupos del equipo científico para diferentes objetivos. Stefan nos estaba diciendo que justamente se utiliza para ver diversidad de bacterias, diversidad de megafauna que pueda estar saliendo de este flujo de agua, así como estudios sobre potenciales virus que puedan vivir también por acá.
I have a question from the background here from Avery. She's asking if we are due for another eruption, and yeah, that's an interesting question because we have evidence for 2 eruptions that have occurred at this site. So the first one was actually soon after the site was discovered, and there were signs of this site earlier on by observations with a camera sled. And then there was an expedition mounted in the early 90s to go back with the human-occupied vehicle Alvin. And while they were diving, they noticed that an eruption just had occurred prior to arrival at the site. So they were not subjected to the eruption. That would have been really dramatic. But they were clearly, just coming month, maybe, or even weeks after the eruption had occurred. So there was lava just everywhere and basalt basically, and all the animals were gone. And then this site was monitored very closely, both from chemistry, physics, seismic activity, as well as the biology, and people were looking at how the site actually responded to this, how animal communities came back and colonized the site, looking at the chemistry, how it changes. And so that was, for some time, it was a site that was heavily instrumented, a lot of cruises, and was one of the best studied vent sites around the world. And then scientists were able to use all the information that was gathered by the chemistry, for example, geology, as well as seismic activity, to predict an eruption. And they were right, actually, and then there was an eruption occurring in 2006. And then subsequently to this, again, scientists were following that event as well. And so based on this information, we might be due for another eruption soon if that cycle actually holds. So we are now like 17 years after the last eruption, so it could happen anytime.
So we should not beat the rocks too heavily.
But yeah, we don't have any signs for it to happen right now, but yeah.
Stefan nos acaba de contar algo sumamente interesante. Acá en la sala de comandos, en la parte de atrás tenemos unas sillas donde todos podemos llegar y ver lo que se esté haciendo, y justamente en una de las sillas está una de sus estudiantes de doctorado que se llama Avery, también del mismo instituto en el que trabaja Stefan. Y Avery le preguntó que si estamos esperando, que Stefan está esperando a que haya una erupción volcánica. Y él justamente nos contaba la historia de que en este sitio han pasado dos erupciones volcánicas. Una justo después de que se descubrió, que fue en los años 90, en la primera parte de la década de los años 90, con el submarino Alvin. Y que justamente después de esa expedición en la que se descubrió, se hizo otra expedición un poco después, y cuando se regresó ya no estaba el ecosistema de la ventila hidrotermal lleno de animales, sino que lo que estaban eran un montón de basaltos y de lava, porque justamente llegaron después de que hubo una erupción volcánica. Entonces, eso fue una oportunidad para que todos los investigadores se centraran en este sitio y estudiaran cómo es que se empiezan a desarrollar o que se empiezan a colonizar estos ecosistemas, ¿verdad? Entonces estaban hablando de que en esa época hubo un gran boom de interés por estudiar esta zona, y por eso es una de las zonas más estudiadas de fondos marinos hasta el momento. Dice que se hicieron estudios biológicos sobre diversidad de animales, químicos, sobre los procesos químicos que se daban en el agua, así como estudios geológicos. Y que en base de toda la información que se recopiló, se hicieron estimaciones y se predijo que iba a haber otra erupción volcánica en la zona. Y las predicciones fueron correctas, y en efecto hubo una erupción volcánica en el año 2006. Dice que en base a toda esta información se ha pensado en que probablemente sea esto unas actividades cíclicas. Entonces, si el ciclo se repite, porque ya llevamos como unos 17 años a partir de la última erupción, si el ciclo se repite, entonces estaríamos esperando una erupción volcánica pronto. Pero no hay evidencia sólida de que vayamos a tener una erupción volcánica en este momento, ¿verdad? Y luego entonces acá otro compañero del equipo científico hizo el chiste de que tal vez no deberíamos golpear las rocas tan fuerte.
Yeah, so now we are proceeding. So we are pumped now for 35 minutes. So we should have enough water in the bag. So you can see it actually filled up. I'm not sure the camera is on it or not. And so we're now proceeding with the wand still in the vent to turn off the pump to make sure that no seawater is coming in while we actually turn off the pump. And then once the pump is turned off, we will remove the wand and then proceed with our operation. I just want to mention here also, like you have seen rocks everywhere, of course, and you might have noticed in some cases the rocks are actually colonized. They have animals growing in them. Sometimes they have biofilms, like you see this whitish to it, so colonized by microorganisms. So we are also looking at what types of organisms colonize the rocks under different conditions. And that's something that a student here, a PhD student from the HUI MIT Joint program, is doing, working with me, Avery Fulford, and she might be able to talk a little bit about that work right now.
Hi. Bueno, en este momento se acaba de cerrar la bolsa de agua. Ya terminamos con la toma de esta muestra. Podemos ver que ya la bolsa está llena. Lo podemos ver acá en la esquina superior derecha. Y justamente después Stefan nos estaba contando que parte de los estudios que se están haciendo acá a bordo es con las comunidades microbianas y con la colonización que hay sobre las rocas que están presentes en esta zona. Entonces ahora Avery, que es la estudiante del programa del MIT que está en el laboratorio en el que trabaja Stefan, nos va a contar un poco más sobre su investigación. Entonces escuchemos a Avery.
Hey everyone, my name is Avery Fulford, and like Stefan said, I'm a first year, just finished my first year of my PhD program at the MIT HUI joint program. And I'm just going to talk a little about what I'm doing on board the Falcor 2. I'm really interested in the activity of microorganisms living at the vent. So microorganisms, when they're in the laboratory, are often growing under really optimal conditions, really high temperatures with plenty of food. And in real life, and especially in the seafloor, where there are these really great fluxes of how much energy is available, what the temperature is, what the salinity is, that kind of thing, microorganisms can really live in a variety of different physiological states. And so I'm really interested in picking apart through several different methods sort of the relative activity or dormancy, basically, of these microorganisms. So by collecting water samples and rock samples that I actually then use a hammer and a chisel to chip away at and then crush up to access the microbes inside, I'm basically using different assays to look at members of the microbial population that are basically completely active, that are metabolically active, so they're doing these redox reactions to, fix carbon and make food for themselves. They're transcriptionally active, so they're turning DNA into RNA and then hypothetically to proteins. And then the ones that are potentially forming these really hardy physiological states. So bacteria that form endospores, for instance, which are these really hard cells that are able to withstand high temperatures and, outside the seafloor they can withstand really high radiation and desiccation tolerance. And then also some microbes that might actually be dead with a collapsed outer membrane or a permeable outer membrane, but are somehow preserved in the environment. For instance, sometimes in biofilms, these cells with non-intact membranes can actually persist because they are in these conditions where a lot of cells are overgrowing them of their same species. So that's an overview of what I'm doing on board the Falcor 2. Let me know if you have any questions.
Muchas gracias a Avery por contarnos sobre la investigación que está haciendo, que es sumamente interesante. Entonces, bueno, Avery nos contaba que ella está en el primer año de su doctorado en la Universidad de Harvard, perdón, en la Universidad del MIT, y que ella lo que está estudiando en este momento es la actividad de los microorganismos que viven en estos ecosistemas. Entonces, ella nos decía que muchas veces en el laboratorio, cuando se estudian microorganismos, los organismos están viviendo en características o bajo características más bien óptimas para su crecimiento, con bastante alimento, con la temperatura que les gusta, etc. Pero que no siempre es así y que en realidad esos organismos en la vida real, en los ecosistemas en los que viven, están expuestos a diferentes variabilidades, a diferentes temperaturas, diferentes límites de nutrientes, etc. Entonces, a ella lo que le interesa es ver qué características o qué habilidades tienen los organismos que viven en estos ecosistemas que estamos estudiando acá. Ella nos contaba que en todo su proyecto del doctorado está tomando muestras de agua y muestras de sedimentos o de rocas para estudiar los organismos. A bordo de este crucero se están tomando las muestras de roca y ella nos contaba que tiene que agarrar los basaltos, romperlos, pulverizarlos, que la verdad ha sido bastante difícil. Acá la hemos visto estar con un martillo golpeando las rocas para pulverizarlas y es un trabajo físico duro. Pero bueno, ella llega, pulveriza las rocas y estudia justamente las actividades de las comunidades microbianas que viven dentro de estas rocas. A ella lo que le interesa es ver cuáles bacterias, o más bien si las bacterias que viven acá son capaces de fijar carbono o si son metabólicamente activas, es decir, que si están expresando su ADN o no. A ella lo que le interesa saber es si las bacterias están pasando de ADN a ARN y potencialmente de ARN a proteínas. También le interesa saber cómo es que estos organismos llegan y se convierten en, bueno, más bien producen esporas o producen estructuras de resiliencia que si llegan a la superficie entonces pueden estar protegidos de radiación o de cambios de temperatura. Todo esto implicaría que los organismos que están acá están activos, pero preservados dentro de las rocas en lo que son los biofilms que producen otras bacterias que están creciendo alrededor. Entonces es sumamente interesante el doctorado de Avery.
Okay, so now we are proceeding at the same site we just were with collecting Riftia.
En este momento, Stefan acaba de decir que ya terminamos con las colectas en este sitio, así que vamos a movilizarnos a otro lugar para hacer las colectas de Riftia necesarias para otros experimentos que se hacen a bordo del barco.
So now we have put things away again, the wand and the pump has been turned off, and now we are proceeding in collecting Riftia and mussels from this site. So the Riftia here seem to be a good size for some of the incubations that are being done on the ship, to put the Riftia when they come back to the surface under pressure again, and do different types of physiological experiments to put them under different conditions and see how they respond, and then look at their transcriptional activity and also metabolic activity over time. But for doing these experiments, the Riftia need to be a certain size so they actually fit into the pressure vessel, which has been a challenge on this cruise because, as mentioned before, the site has now been not subjected to volcanic eruption for a long time, and so the Riftia that are here get bigger and bigger. But of course there are new ones coming in as well, so there's some smaller ones, and we are hoping that the ones we find here are actually of suitable size for Jessica to do her experiments.
Stefan nos
So we would
estaba
actually put them in the... Yeah, okay.
Stefan nos estaba contando que, bueno, sí, ahorita vamos a muestrear los gusanos Riftia y nos contaba que justamente lo que se quiere saber con estos especímenes es cuál es su actividad metabólica y su actividad transcripcional. Más que cuáles, es ver cuáles son los límites de esta y cómo cambiaría bajo diferentes condiciones. Y para poder hacer ese experimento se necesita que los gusanos sean de un tamaño más pequeñito o mediano, y como no ha habido ningún tipo de disturbio en el ecosistema desde el 2007, esos gusanos han crecido bastante. Entonces en el sitio podemos ver que la mayoría de los especímenes son muy grandes, pero aún así siguen creciendo, nacen especímenes nuevos, por decirlo así. Entonces aún podemos encontrar gusanos del tamaño que se necesita. Es cuestión de buscarlos. And Stefan?
Yes.
There was another question in the chat about how violent are the eruptions in these kind of sites. Like the ROV would survive one of these?
Yeah, that's a good question. All I can think of is that it will be a massive force that will be happening here, like lava actually, of like over 1,000 degrees centigrade coming to the seafloor, interacting with the very cold seawater. So there's going to be a lot of force. So I don't think you want to be close to it for sure. But maybe, I don't know, it's hard to tell, like, how from how far away you would be able to safely watch it. So I think if you have a robotic vehicle, it might be possible, to watch that potentially, but
nice footage.
But it's hard to tell.
Estefan nos estaba respondiendo acá a una pregunta del chat de qué tan violentas son las erupciones que se dan en estos sitios, y nos estaba diciendo que justamente él cree que sería una fuerza grandísima porque estamos hablando de magma que está a más de 1000 grados centígrados de temperatura reaccionando con agua que, como podemos ver en este momento, está a tan solo 2 grados centígrados, un poco menos de temperatura. Entonces sería una reacción bastante violenta, que no tiene con seguridad un número de a qué distancia sería algo seguro para ver estas erupciones si uno está utilizando un ROV como el Subseafloor en este momento, pero que podría ser posible. De hecho, un dato interesante es que si uno se pone a leer literatura sobre este sitio, hay varios experimentos que han sido registrados que han sido cubiertos por magma. Los científicos llegan y ponen sus experimentos acá, pero si hay una erupción y hay magma, entonces los equipos y el experimento en sí termina cubierto y embebido en la roca. Entonces, algo que sucede al momento de estudiar estos ecosistemas.
Okay, I don't need 2 grams, no muscles. Jesse maybe wants 2 grams, but I don't know. Muscles we don't need.
Okay.
You don't need muscles, but even from the associated one.
Oh, an octopus.
Podemos ver acá un pulpo en la parte izquierda de la pantalla.
It's for Sabine for the small fauna, no?
Okay.
I don't need 2
Okay,
grams.
so we'll grab some if you want.
Ayer vimos uno igual, justamente que estaba escondido en una de las grietas de la ventila hidrotermal.
All right, yeah. Can we get? Okay, like maybe get Jesse.
También podemos ver encima de las almejas que hay un camarón de profundidad.
Yeah.
So maybe here's a weird question. All of these sites have been described and named just in like the past 16 years. If there was another eruption of that magnitude where it kind of scoured everything clean,
yeah,
the new vents that arrive, like those then get named. You're not going to be like, this site used to be crab spawn, right?
Yeah. No, that's a great question. I mean, I think though it's there are definitely sites that actually survived the eruption. So the animal communities were wiped out, but in some cases the hydrothermal activity remained. So especially the high temperature structures like PVAN, Bio9, where we might go later on this cruise actually, or on this dive, they remained. And and some inactive sites too that still have the name, like MVAN for example, and some other sites too. And so it all depends. If a site actually remains active after an eruption, then I think the name, if the location can be identified or confirmed, then that name will remain. But there are probably going to be a number of different sites opening up as well.
Wow. Okay.
Yeah.
Acá uno de los técnicos del barco le acaba de preguntar a Stefan algo muy interesante, y es de que si hubiese una erupción volcánica en este momento, entonces tendrían que cambiarle el nombre al sitio, porque vieran que los sitios que están por esta zona todos tienen un nombre específico, y ese nombre es basado en coordenadas que los científicos apuntan de dónde es que están las ventilas hidrotermales. Pero si hay una erupción volcánica y la ventila hidrotermal se cubre, entonces habría que cambiarle el nombre al sitio. ¿Cómo sería eso? Y Stefan nos estaba contando que justamente cuando hay erupciones o cuando hay actividad volcánica nueva en los sitios, las ventilas hidrotermales suelen aguantar esto y suelen quedarse en el mismo lugar. Entonces, en realidad no habría que modificar muchos de los nombres, pero en caso de que una actividad volcánica le quite o modifique demasiado el ecosistema, probablemente sí se tendría que modificar el nombre, porque ya no habría una ventila hidrotermal en esa zona. Entonces, parte del dinamismo que tienen estos ecosistemas que es muy interesante señalar.
This and left now, Stefan.
Hmm?
Almost to the left of the screen. Left.
Yeah, right
There.
there. Yeah.
Up.
Yeah, the
Up
plume
and
and
left.
between.
Oh, here?
Yeah.
Too high. There.
Yeah.
Okay.
So you'll be.
So let's go for this one first, right? Maybe.
Right. I'll just clear the whole space.
Acá pueden ver que la biobox grande que estamos utilizando en esta inmersión es una hilera, y es justamente porque el biobox que se tenía diseñado para el ROV no estaba siendo lo suficientemente eficiente en guardar la temperatura del agua al momento de llegar a superficie. Entonces los organismos estaban llegando muy estresados y algunos incluso no podrían sobrevivir todo el viaje. Para los experimentos que se están haciendo a bordo se necesitan que los organismos estén vivos. Entonces se decidió cambiar el diseño y poner esta hilera a modo de un biobox grande para poder colectar organismos y que llegaran vivos. Justamente ayer fue la primera vez en que se utilizó la hilera a modo de biobox y fue un éxito. La temperatura del agua en superficie fue de solo 6 grados, que claramente es una diferencia bastante grande con la temperatura alrededor de las ventilas hidrotermales, pero es suficiente para que los organismos sobrevivan.
That's almost too small, huh? That's good. Okay. So let's try to find a couple of more.
Y ahí va el
This
primer
size.
gusano colectado.
One is it here?
Uno de los equipos a bordo que experimentan con estos gusanos son el equipo de la Universidad de Harvard, y ellos son los encargados de hacer los análisis de fijación de carbono y de transcripción de estos organismos.
Is my mic on? Oh, yes. So I'm collecting tube worms for my high pressure respirometry system, and which is basically built into a shipping container that we put on the deck of the ship, and it's used to keep hydrothermal vent animals alive at in situ conditions. So in situ conditions meaning slightly warm water. So we run it around 15 degrees C, and we flow the vessels that are under high pressure with hydrothermal vent fluids, so sulfide and oxygen and a lot of CO2. And I use the system to be able to control the environmental conditions to look at metabolic changes in both the host and the symbiont. And it's really challenging because my vessels are quite big for pressure vessels, but even so, I still need worms that are only about a foot long or 30 centimeters, depending on what part of the world you're from.
Acá la doctora Jessica de la Universidad de Harvard nos estaba explicando en qué consiste el experimento que ellos tienen, y es muy interesante porque ellos están trabajando a bordo, pero no en los laboratorios del barco, sino que en uno de estos contenedores que se utilizan para transportar mercancía. Ellos lo modificaron para crear un laboratorio móvil, y dentro de este contenedor, de este laboratorio móvil, es que están haciendo sus experimentos. Entonces nos contaba que lo que están haciendo es ver la respirometría de estos organismos bajo situaciones de presión. Justamente ellos tienen unos contenedores que son presurizados y en los que bombean fluido hidrotermal con con oxígeno, con azufre y con dióxido de carbono, que es básicamente haciendo o replicando las características de estos ecosistemas. Entonces es muy bueno porque están haciendo unos experimentos básicamente in situ de cómo se hace la transcripción y fijación de carbono de estos organismos. Ella nos estaba contando que este sistema le permite a ella controlar las características ecosistémicas en las que se desarrollan los organismos, y puede ver cuáles son los cambios que tiene el gusano en sí, pero también los cambios que tienen las bacterias simbiontes que viven dentro de estos gusanos. Y con estas imágenes de colectas de gusanos Riftia, termina ya esta sección de narración en español. Acá los dejo con las imágenes que tienen sobre los pilotos colectando los gusanos, que Jessica justamente nos estaba diciendo que necesita gusanos de aproximadamente 3 centímetros, un poquito más grandes tal vez, para que puedan entrar dentro de sus contenedores presurizados y poder hacer el experimento. Entonces acá los pilotos siguen con la colecta y el live sigue. Recuerden que tenemos tiempo en el fondo aproximadamente hasta las 4:30 de la tarde, hora Costa Rica. Así que seguimos acá explorando el fondo marino, y se queda también por acá el científico Stefan Schwieber, y si tienen más preguntas pueden ponerlas en el chat y él las estará contestando, igual acá con los demás técnicos y pilotos. Así que seguimos.
I wonder if you just scoop the whole clump. And there might be small ones. These guys are too braided, huh? They can't be pulled up with the...
Just yank the whole thing up.
'Cause I think these guys over here might be options. Like that probably is too big.
Oh, that's too big, yeah.
Yeah.
Now you want to go to the right and see this kind of grab. Watch and see what. See the little thing up to the median.
Yeah, you can put that in and then.
I think we try to.
Yeah,
see
it's
this.
not gonna be.
Yeah.
How far away is P gun?
I don't know. Like 20 minutes. I need to move.
There's some small ones right there. Below us.
Yeah.
Oh, one small fish.
All right, well, thank you to Yvonne
Oh, yeah.
for the past hour or so here.
That might work.
I will try and then we can move on. Yeah, these are good.
Quick comment for anyone who tuned in yesterday, and I know folks were kind of like, is that a cooler on the front porch? It is. It is a cooler. Did it work? Yes, it did. It kept everything in there nice and cool, as it's designed to do, I suppose. So another item that you can just buy off the shelf that happens to work. Now we did need to modify it, right? Because these are double-walled, these coolers, which means that there's a gap in between the outer wall and the inner wall, and it is not designed for the pressure of the deep sea. So we had to drill holes from the outside, from the outer wall, into that gap in the middle. So it still holds water on the inside, but the spaces inside, the gas inside the those walls there can get compressed just fine without crushing the rest of the container. But no, it worked well. But it does almost seem like Sebastian's just going for like a day at the beach when we open the cooler up.
I think those are going to be a good size.
A good size for you, or a good size like big?
Right there, yeah.
Yeah.
That, come on, go in.
So the way they've been collecting these worms is you have to kind of bump the gills there to get them to go down in their tube, and then we're gripping the tube where the worm is no longer actively in it.
Hopefully it doesn't have a big long tail.
And we try to then pull it up off the substrate. But if we grip the tube where the worm is, then obviously that will smush the worm. So it's difficult. As you can see, the tube isn't super strong, but we've collected a few this way now.
Party at the bed. Looks like confetti. What a pretty sight, though.
There it goes. Nice. But it's long.
Well done, but how long?
No, it's not bad. Nice, well done.
But look, I think that a lot of that is empty tube in the bottom.
Okay.
I think that might be good.
You want to try the ones on top?
Yeah.
Do you kind of candle the tubes in front of a light when you get them to see where the worm is? When you get the worms up to the surface, do you sort of shine like a light through the tube to kind of see where they are?
I don't really need to. I kind of just, you can see.
Oh, you
yeah,
just see
and
right
then
off the bat.
you can kind of pinch their.
Feel a little bit?
Yeah.
Okay.
And then if they've got a really long false bottom, I like just zip tie it. I make a.
Go on.
I cut the bottom and then zip tie the bottom so they don't fall out. And put them in the pressure vessel.
What are the tubes made of?
Chitin. That's a slightly different type of chitin that I know some people are working on that as a potential use in material science, but I don't know.
Yeah, it's a combination of like chitin and protein. Alright, go for this one and then the other one back there. And then we move on.
This one, so that gotta be middle, and then go behind the arms.
Ooh, that might be too long.
Question in the chat about whether or not the pilots have a perceptible lag between their control input and when they see things move. No, not really, not on this setup. It's quite impressive. We're pushing the control signals down 6 kilometers of umbilical, and then the ROV is doing the command, and we're getting the footage back up the 6 kilometers, but it's all fiber optic. So roughly the speed of light. It's not perceptible when we're doing it. And I've just messed with the manip a little bit. They've let me kind of move it, and it's an odd feeling watching it because it does move the second you move your hand. It's very cool.
We have to see me turn this back up and cover this.
Yeah, and there's that one too.
It's too close in without.
Yeah, there's several fibers down the umbilical.
You won't wash out the small one down here. I don't think we have to reposition it up there.
Okay.
I mean, it's close by. We might still be able to go back to Purpose Bar. So let's pack it up and then go to dump those.
It's about all done here, don't that one?
Yeah, we can maybe. Yeah.
I have one last one.
Uh-oh.
Whoa.
Those aren't even limpidine muscles.
No.
I wonder if there's any babies in there. I don't know if that's gonna be.
Okay. Alright, pack it up.
Where are we going next?
South to Bio9.
How much time do we have to sample at Bio9?
We're gonna have to do some slurps for Sabine, and then in Riftia's potentially P-bend. So depends, like, you know, when we get there. I'm not exactly sure.
Because I think that the small two rooms take a little bit of time. That's the problem.
This is south.
Looks like we've got about an hour and some maybe.
Camp's good.
We'll be hopefully bringing you the ascent again today. That'll be over on YouTube if you're watching on Facebook. We'll finish up on the bottom, and then if you're interested in kind of hanging out with us all the way back up through 2.5 kilometers of water. That'll be over on our YouTube channel. But we have been finding stuff along the way. Had some gelatinous critters yesterday, but we've also been seeing shark pretty regularly just in the last 100 meters or so. Even less than that. We've had some oceanic whitetips up there. Also had what looked like maybe a Humboldt squid briefly go past us yesterday.
South.
They must be in the coordinates, I think.
Looking back at our connection data, it looks like stream's been pretty good today, but the weather's also been pretty good. While we're repositioning here, I'll just toss up our outer view.
No, it's further south.
A stark difference from what we've had the past few days. Real nice weather out there.
Yeah, I think they should be on. Monica, do you have the coordinates for Bio9 and Event? I don't see them on the. Oh yeah, here we are. So Event, here we are. There you go. I thought they were already on the map.
So Bridge Avenue waypoint for you due south.
Yeah, copy that.
Oh, there we are. Event. Okay. Oh, you just entered it. Okay.
Do you want the Bio9 as well?
Yes.
Going south.
Okay. Okay, let's go to P-Vent first. Yeah, but I think Sabina wants to have connected confirm with her, but I think Alvanella, Riftia, the slots in Riftia is what she prefers. In Riftia. Event is Riftia. I think that's all mentioned. Okay, we're passing Bio9 anyway first, okay. Check it out.
I can't go anywhere just yet, so this ship is too.
Right.
So, this is crazy.
Good job.
Thank you, okay, thank you.
Should I make a biological observation on this Bio9? Stefan? Should I make a biological observation on this Bio9?
No, this is not Bio9. We are far away from it right now. But that's, I mean, not too far away, but.
There are last groups we found a loop at the cable.
So we have to wait for the ship to move right now.
that, yes. 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 Good.
Bridge RV, that's update waypoint. What's speed are you doing right now? You could probably get up to 0.6 because we're heading about 120 meters. I'll give you a shout before you slow down.
No worries, as long as you get me a final waypoint and it's not like oh, it's got the other kind of thing, it's fine.
No, sounds good.
That.
Bridge RV, that's your final waypoint at the bottom of the chart there.
All right, so we are just waiting on a little ship move here. To show you sort of what we've got. We're wanting to go visit those two yellow points down there. And we are currently just above that red circle, but the ship is now moving to the sort of cyan blue dot in the lower right. And so we just got to wait for it to scooch along and we'll sort of follow after. There you go. And Russ zoomed out a little bit and you can see Sebastian at the very top there. Falcor 2 with the green trail now showing the movement. And we're just going to follow along after.
Fit channel and then come back out of there.
That one's one of these things.
Another beam trying to fly into it. 3 or 4 caves. Vertical.
Okay, we're getting close to Bio 9, which is a high temperature black smoker. We can actually see it coming up here right now. So you can already see the black smoke coming out. So this is really super hot hydrothermal fluid, undiluted. So this is really what forms deep down in the Earth system and comes out undiluted at very high temperatures. So this is probably around 360 degrees centigrade. Just a guess right now based on previous cruises, but It's super hot fluid that's acidic, like pH around 4, high in metal content, and very reducing, so it has a lot of hydrogen sulfide and hydrogen gas. And when this comes in contact with the seawater, all the minerals or the metals that actually are in solution at the high temperature and other reducing conditions precipitate out and make the appearance of the black smoke.
So this is bio-9?
This is bio-9, yes. So we can make the observation, or like a frame.
Get up out of here. What's that?
Have you dropped in?
No. I built it behind stuff. I don't really want to do it.
So we are now at bio-9, and so it's covered, I think, with albinella. We can see the like white stuff, but it might be a little tricky to position it. So it's yeah. Yeah, now we are
further south.
a little further south, but really close, yeah. So I thought, like, you wanted to have a slurp in the albinella, maybe at bio-9.
Yeah.
It's possible.
Yeah.
So we are just in the smoke right now, so we need to reposition. But yeah, here we are. No, that's not it. Yeah, that's a different.
It's a new one? Yeah,
That's also part of bio-9, I guess.
Yeah, I mean, there's some other like venting structures, but it's not. So we need to go back to bio-9. Yeah.
Back?
Yeah.
Oh, back there. Okay.
Oh yeah, yeah. No, we. Yeah, sorry.
I thought we were going to feed that.
Oh, sorry about that.
That's fine.
Yeah, we set it to the furthest one.
Sorry. Yeah.
Yeah.
There's some rift gas potentially down there. There are also some experiments. I'm not sure. Yeah.
It's spinning off to your aft towards the ship. So it's outside.
So there's bio-9 now. So there's not really that much at the base, right? And then you see all the white. It's probably albinella. If it got closer, maybe at the
That's a huge
bottom.
structure, huh?
Yeah, it's really big. Yeah.
I had some comments about our audio getting desynced, so just doing a time check here at 21, 28, 38.
Closer at this lower structure here and see. Maybe here.
Maybe I have a good picture from the bio.
Hmm?
No, just that
Yeah.
I have frameworks that you can.
So yeah, I actually go up here. I think this is quite nice here.
For how high is it, huh?
Yeah, it's like 10 meters or at least or something. Yeah. It's an impressive structure. Yeah. So we have to be careful, of course. There's some hot water coming out here.
Yeah, we're gonna have to do some sampling here, Tony. So we just hang right here. We're good.
Did Max put that there?
So I think this could be. What do you think, Sabine?
To zoom in a bit.
Give us a little.
Oh, it's crazy,
Oh,
huh?
okay.
It's Is it possible to sample there or not really, because you cannot touch base? You know what we do? We make a biological observation that there is albinella here, and probably we first check out event, and then we can go back.
Okay, should we look around a little bit if there's anything else? Okay, just maybe fly around
See
it.
a bit the area.
And so we make an observation that there's Albanella at bio9.
Boah, that's massive, huh?
Oh, wow, that's huge.
Yeah, it goes even higher.
Yeah. Phew. Can you make some highlight fragrance fiber from this nice views?
The black towel.
Can you see sorrow too?
Yeah, you see here like it's really like hot fluid coming up.
Yeah. It's crazy, huh?
Yeah.
Alright, I'll stop at this.
I mean, this looks like the most active kind of Albanella colony.
Yeah.
Right. Okay, let's move to P vent.
Do you want to sample here, or do you want to?
Yeah, not for now. So we made the yeah, we might go back, but we'll check out P vent next. So this site here, Bio9, existed actually prior to the eruption in 2006, and it remained active through the eruption, as well as P vent, which is the next high temperature structure that we go to, Black Smoker.
P-vent got inactive in the beginning and then reactivated again.
Oh, not that I know of.
Yeah, because we were there, we had like experience there at P-vent, and it like was inactive in 2009.
Mm-hmm.
So it was active in 2000.
Is that a fauna?
Alright, if you're just joining us, we're currently coming to you live from 2.5 kilometers below the surface of the Pacific Ocean on the East Pacific Rise. We are south of where we've been for previous dives on this cruise, and we're headed past, or we just headed past a real nice black smoker chimney vent. This is a hydrothermally active area. We've got a little while yet on the bottom, but we are towards the end of bottom time. Fun shrimp in the water column there.
I think that's it, yeah. doesn't need to be in a rift, yeah. Definitely could be used for the warning. What do you want to do? Yeah, here's the really big smoke coming out here. Oh, there might be.
But there's a marker there.
Yeah.
I think that might be a high temperature logger, I'm not sure. You think that's rift here? Is that rift here?
No, I don't think so.
Can you take a look? Yeah, that's a very massive black smoker down there. Venting really vigorously. Yeah, and what you see here actually is a temperature logger that has been placed by other scientists to measure the temperature over time. So that will be put in a chimney, and then it will grow with the chimney, and then eventually it will be recovered, and then the temperature can be traced back over time. Yeah, no rift here.
No.
And we should not really mess with this anyway.
There's something to the right.
There's some empty tubes here, I guess.
Yeah. Oh, not gonna.
Yeah, from a biology point of view, it seems like maybe Bio 9 was more. Can we go here maybe? Hmm? Oh. Yeah, I wanted to.
Yeah, just for a quick
But
business on the way.
I don't think there's anything there, right? So yeah, maybe yeah, check it out. Yeah. Yeah. I think that was the site that used to be active,
Yes.
where Lauren had sandwiches.
Yeah, and
I
also we
remember
had
this.
the sponges there.
Yeah.
Can you zoom in a little bit?
Yes, of course.
So that's old dead Bacterial shells.
Yeah, wow.
Can you make highlights?
What is that?
This is P-vent, and this is Bacterial shells.
Marker O actually. O marker. Bucket lid marker
Because I
O.
think the pictures that Monica had last looked at was like also still had rift here, so probably in the meantime.
Yeah,
we have
no, that used to be quite.
yeah, muscles. And
yeah,
now
right.
You see the, it's.
Yeah.
Well, it's very dynamic, huh?
Yeah.
Within like a few years, you get a change of like.
Yeah.
It's super dynamic.
It was already dead in December.
Yeah.
But you mentioned you saw like riftia at the base of P-band or something?
There was some on the chimney we just flew over earlier.
Which one? Bio 9?
Halfway up the chimney there were some riftias.
Really?
Yeah, they're hugging the alveolas essentially.
At Bio 9?
No, P-vent.
Okay, didn't see that. Okay, let's go back to P-band.
Sorry, where are we now? Is it by
P-band.
Here, P-vents.
Okay.
How did it look?
Or
So
the
in
chimneys. I mean, they were on the chimneys.
Yeah, I think you might be talking about Bio9. Let's see.
No, I know it's not bion.
Okay. So here, there's nothing, right?
Not this chimney, the big one.
Yeah.
We could cook an egg here.
Yes.
Scientists are just trying
definitely
to
this would be a good place for that.
figure out where to spend the remainder of the dive, it sounds like.
Yeah, right, no. Anything up here?
Fly and see.
Sorry. It was there a few here and there, but if you have to either come closer or zoom a lot more. Go closer. There was at least one big clump on one side of this chimney. These albuminal tube prints, imprints.
But doesn't seem to be active Salmonella colonies.
There to the left of the chimney. So this. Down and left. Top is unlikely. Where are you gone? Yeah, it looks like little chimneys and not too worth there.
Currently on the lookout for some more tube worms, perhaps.
Get closer a little
Can I have the camera for a second, please?
Yeah.
I'll get closer. I just don't
Yeah,
know
no,
where
sure.
everything is because it's moving around too much.
Yep. Sorry.
No problem.
You know, we just got up kind of just looking up to the right.
Alright, go ahead a little bit. Where do you want to look? The top of this, middle of this, left of this, right of this?
Yeah, no, further up.
Okay.
So we're just going to work our way up this chimney, looking to see if we can find any worms.
Before we would leave this, should we do the here? Yeah, okay. But here is, I think it's quite easy to position maybe because it's like.
Do you want to put the egg next where the temperature lock is?
I mean, if we put it like in the, yeah, maybe not. It depends like how well we can position ourselves, but maybe that's too risky.
Do you want to put it like on the smoker area or?
We first, I suggest that we probably go back to.
So here doesn't seem to be. Yeah, go back to Bio9. What do you think, Stefan? We don't seem to find any.
Yeah, I mean, that's the game with hydrothermal vents, right? You want to find some reef gas, correct?
Yes.
So, well, obviously there's nothing
Yeah.
here, so let's
Yeah.
go back to Bio9.
And then send
But,
back the other.
yeah, there was almost nothing at Bio9 either.
Not Bio9, there was on top like all the other handles.
Yeah, there were albinos on
Yeah,
top.
yeah.
Yeah, that's what I mean.
Yeah,
Nothing
yeah.
as far as
Well,
two worms
yeah,
are concerned,
yeah.
sorry.
Yeah.
these are all remnants of alveolinits, but not really. Okay. We'll have a nice overview of this. Yeah, let's see.
The best thing is that I fly it. It'll be a better plug-in there. Yeah.
Highlight. Okay, let's go to Bio 9.
Alright, so we're going to head back to Bio 9, which is the vent, the smoker that we passed on the way here.
Go to the alveolar. Look for Riftia maybe. Alveolar. We have two slopes on and then do the egg, or measure the temperature, do the egg, and then maybe if you have time, break off a piece.
So Jeremy, so you waypoint for it? Did you
Break
sort
off
of
a
get the
piece.
waypoint for it?
Yeah, we'll see. Probably not.
But it's
the
very
slopes
close.
are more important.
And it's very close, so we can go there again. We're there like within like 15 minutes from Tibet. Then we visit everything. So
Yeah.
it's,
You're right.
So there is no diffuse flow in those maximum covers. That's why there's no light.
There used to be more, but it's really not there anymore, yeah.
You don't use both of these better. Is this one or the other reference?
So these
That's-
both two structures are from.
I think there's like Bio
Bio
9 and Bio 9 Prime.
Mhm.
So let's check this out here first then. Yeah. Should we go for this one? And then we can go maybe do another slip on the other structure. How do you think you could position well enough to do a slurp here?
Yeah.
Okay.
Alright, so we're going to
Let's
be
do it.
doing a suction sampler here. So this is the large hose on the front of the ROV you might have seen a couple times. Going to kind of vacuum up some of this mat on the chimney here. Now, as you can see, the smoke on these, different work environment than where we've been the past few dives.
What's that? What's that, bro? Your board armrest. Board armrest move that side. It usually does.
So these chimney structures can be quite delicate, so pilots are just trying to come in gentle. You can see on the right side there, and again in this camera, they've extended out, and so we can kind of rest on those.
Probably we cannot really sit on there. Either basically likes to fly. If this is not possible, it's not a problem. It's not like first priority sample. It's a nice to have. So if it's not possible, it's not a problem.
Okay. Well, Christopher, when then raise up four chairs and get rid of this hook. It's not private. We're trying to get it fine.
So we would use chamber number 2. We're trying to do, if we can position ourselves, we're trying to do a slow, like a section sample. Yeah, at Bio9. Bio9 South. But it's not clear yet.
Yeah, I can try to come down to the seabed if that'll make it easier for you. We're way up high.
Can put it on number 2.
So as I mentioned, chimneys can be quite delicate, so rather than landing on this one, pilots are going to attempt to do suction sampling off the surface while kind of just hovering at it.
Is the position of the chamber number 2? Okay.
Second, drilling the starboard rail.
So while we get set up for this, if you're just joining us, we're towards the end of this dive, but our last little leg here, we're going to be taking some samples from these black smoker chimneys. These are much hotter vents, much more intense than what we've been seeing on our earlier dives. We are streaming this live from about 2.5 kilometers below the surface of the ocean using a remotely operated vehicle named Sebastian, being controlled from onboard research vessel Falcor 2. And as I mentioned, the pilots are going to be approaching the chimney and attempt to use the suction sampler that you see there, kind of like a large underwater vacuum cleaner. They're going to try and sample from the surface of the chimney. This part of the chimney on the side of the screen here.
Just leave it, just leave it.
Right. You go. Do you want to follow it? Or
yeah, put it up where it was. Yeah, that's good. That should be everything. That's fine.
So there are, you can see some of the red worm extending out. So these aren't the Riftia tube worms that we've been seeing, but I believe these would be Pompeii worms. So we have worms and are doing just some suction sampling around here.
Okay. That's good, great. Nice, okay.
As you can see, the material of the chimney really just goes in there.
No more?
What do you think? A little more?
That's the fine mesh, right?
Yes.
Yeah, it looks already a little sluggish.
Yeah.
So,
Yeah.
Let it run through for a bit. There's a lot of stuff in the chimney.
Yeah, because they're
Yeah, I think it looks pretty.
There's a fair bit in there.
Yeah.
Okay, let's turn it off.
So on the suction samplers, we have some different grades of mesh.
And then maybe go to the other structure. Next.
And they,
what's that? These cores, right?
No, still another fine.
feel that there's enough in that particular container.
Number
we sucked
three.
up some pretty good pieces there. So.
We did coarse and
we have only
fuzzy
two
bear,
cores.
fuzzy rock. And then
Right, we have only
the
two
sugar.
cores, right.
Okay, we can go to the other.
Stamped up.
Oh, it's stamped up. Oh.
Yeah.
Oops.
It's not going to go anywhere. See the turn rule at the bottom? It's out of this thing.
Oh.
So,
Okay.
Sorry, man.
Should we do the egg thing?
Then I think, yeah, I guess you want to collect first the rock.
Yeah, maybe. Yeah.
Sounds like next we're going to get a rock sample
Chimney piece.
Oh, right.
from the chimney.
You,
How much time
I
do we
mean,
have
really,
left?
I'm just
Like
gonna
half an hour?
sample one of those chips and
That would work into one of the quivers.
Yeah. But the question is, yeah, where, which? But ideally we would measure the temperature.
But that takes too long.
Yeah. So maybe do the egg.
Yeah.
And then we have that done.
Yeah.
And then we can focus. Yeah.
Yeah.
You wanted to measure the temperature for that too?
Yeah, let's first cook the egg and then
Yeah,
there's,
okay.
But also you have to say how important the collection figure is, because this egg thing is like a nice to have, you know, it's like
Yeah. But for the, yeah, if we collect the rock, it would be better to actually have the temperature going along with it. So.
Let's
Huh?
first look.
Temperature.
Yeah, I know.
So let's have a look and
Yes.
find a small chimney that
Let's find it,
could
yeah.
be broken off, where we can measure temperature.
And then maybe
just,
do the- yeah, that's a
decide,
good
then
idea.
we decide
Yeah,
first
yeah.
if we need to find a suitable spot.
Yeah, yeah.
For either one or the other, because we cannot be like on top. Must
Yeah,
be
maybe-
small enough.
We could probably land out
Down
yeah,
here?
We could land out just to the left of that.
Okay, that would be great. Yeah, maybe.
Let's see what we can do here.
That's the other structure now, right?
Yes,
Yeah.
the left.
Okay. Yeah, yeah.
Nice flow coming out of there.
That's very active. It looks like an organ pipe. You
Yeah.
know? I mean, further back there might also be a good option, potentially, I'm not sure. Like
Oh,
here.
Okay, I can remember that.
We are still playing around with it's not hit it. We are still playing.
It's massive. Phew.
That you cannot put in the corrector. That you cannot put in the corrector.
No. But you still have a bio box left, or Stefan?
Oh, we still have a bio box left. That's correct. Yes.
Probably
Yeah.
easier.
Thanks. Yeah, you're right. Yeah. Thank you very much for pointing that out. Appreciate that. It has been a long day. That would be much easier for sure.
Yes.
Maybe we can even park it and then.
Nothing right there.
Yeah. That would be perfect.
Let's set up for that.
Is it bungee off that one?
No.
Let's get bungee off that. Whoa.
Looks like watch that. There you go. Hold on. Stand by. Alright. You have a visual on that, so we set it a little bit. There you go. That's clear. What's that?
I think we're going to run these out.
This is the one over here.
There. Oh, it's in the back now, so it's in the back.
Yeah, probably it's harder to get to. I think this might be easier. But it's a big one. Maybe this one here.
But they cannot get in.
Yeah.
We could get kind of in.
We could end up damaging them.
Yeah, no, maybe that's, yeah. So this is, can you get to this?
Yeah. It's just the porch right underneath it.
Basically just touch on it.
Yeah, don't put the porch right underneath it.
Kind of square.
And just need to be able to see the edge of the box, that's all.
So square.
Alright. You can knock that in?
I was hoping you'd be
I mean, not this whole big thing, right? You're not talking about, I mean,
I think if I go up and
talking about something like here, up here.
Alright. Hold on, I'll come up later.
So again, just trying to figure out where to collect from. Trying to collect a rock sample from this chimney. Chimneys are quite brittle, quite fragile, so actually getting the piece of rock off of it won't be too difficult. More a consideration of limiting how much additional rock we take off of it.
I mean, from here as well.
Yeah, that's kind of what I'm thinking.
No. Alright.
Open the box.
Slowly. It's 3.
So this one is probably, which one are you shooting for? This one?
I think so.
Yeah, but that's probably not as active. I mean, there's not that much coming out here.
Alright, so that's all the way up.
You know, one to the right looks pretty good, Stefan, right?
You mean this one? Or this one?
yeah, go on
This
one.
one, yeah, no, this one would be great, yeah.
But I'm sure the other ones will be plenty hot for the egg anyway.
Yeah.
I think we'll go out to your report on.
Not for the
Go
rock.
ahead,
Who
Okay, alright.
because I know it's going to be hot.
Yep.
I'd like it to be softer so I can.
Sorry, I was resting with the egg in my mind to cook it.
Alright.
I understand the limitations of this situation.
Hold on, maybe on this view I can.
You guys are aware that these may melt your plastic fingers, right?
It wouldn't be the first time.
Okay,
Yeah, we've done that.
Okay. Don't want you guys to be disappointed.
Just printed off the 16 and I was going to get
Maybe a little lower.
I think it's pretty wide in that direction.
And very gently, I think this will be very fragile.
I'm thinking about that.
It stays.
That's nice, that's nice. That's good. Yeah, yeah, it's perfect. Wow, great. That's amazing. Yeah, yeah, yeah.
You're above.
No. So as you can see, quite brittle that.
Maybe some in there.
Yeah, maybe a little bit, but
Can you zoom into the dialogues, Chris, to see if some fell in there?
oh, on top of it.
It's upside. It's upside? Yeah, it's.
Can you open any further?
No, it's all Scott.
There you go.
Okay.
So close.
It's push on something, but yeah, that's all it is.
There's a piece here.
There's some pieces in the bottom. If you look in the bottom of the box, there's a few
Yeah, I see
Okay.
some
segments. But
Okay.
Let's see if we can do that.
It's already good. Like a little more, but it's already good.
Oh, I guess
Do you want to wash it?
Yeah, we could try. I mean, like, yeah.
I cannot really grab it.
I don't know what this is. What do you want? I'll get whatever you want to get. Yeah, which one do you want?
Yeah, where can we go? I mean, maybe try again here, but this will be very fragile, I guess.
that's what I was looking for.
Oh no.
Right, it's not that. It's too soon.
The whole thing's moving.
Yeah.
Just leave that one.
So yeah, I think both things wobble.
Yeah, yeah, I know. Yeah, I know. It's very, yeah, it's very dicey.
Yeah, I think if we go in again, we're going to end up knocking off completely. We could try the port arm and see if we get that one on the other side.
I mean, we could, yeah. I mean, there's definitely also hot water going through this one, but
Do you want to try that one then?
yeah, if it's easier to get that. And then maybe we can measure the temperature once it's removed. There might be some additional.
Well, we know it's above 220 then.
See, we don't need a thermometer, a tea probe.
That's what it melts at.
Super soft. Yeah, it's like carried over.
Yeah, no, another melts here.
If you were to, I don't think it's gonna collapse.
We have like 15 minutes. We should probably wrap up at some point. Do we want to do the egg?
Probably can hold the egg and then
Yeah, and then
wrap
leave
up the
and then wrap up. Yeah,
Yeah.
I think that's what we need to do.
The egg.
I think it's already in the scoop,
It's
right?
in the scoop.
Yeah. So let's close the bio box. I mean, we might have something in there that can be used, hopefully.
I create sample before with the rope in by the
Right.
Stefan.
Give me a view of that.
I don't know what we're talking about.
I create sample.
Oh, that's that little one. That's what that was.
I create sample before in the box 3 the rock. Correct, Stefan?
So that was sample number
The
84, right?
This.
Yeah, it's a small one.
Can you make a video highlight?
I will do.
Okay.
What would be the effort to measure temperature?
Should get over there.
Could we go there right now, potentially?
You think you can freeze that in there and then get the temperature probe out, or is that?
Yeah, I could try. You won't have to.
All right, I'm gonna march in.
Hold the door.
Oh yeah, that's a bad idea.
I mean, is that okay? Or you want to keep it or take it out already?
Just discussing like also what is good for the arm of the ROF, I would say. I don't know if it matters that the arm is now.
It's not very good for it now.
It's not very good for the arm, I think. If it's like,
Not
like
really,
this.
no.
Okay, then maybe.
How hot it is?
Where is the arm? Do you have you?
It's above there.
Yeah.
Yeah, maybe take it out.
Yeah.
And then probably has been.
It's now 1 minute about.
No, not yet probably,
Yet.
but.
Watch out.
Or probably you can hold it a bit like that the arm is not inside, just the yeah.
Yeah. I'd come over and spin.
What's that? I don't kick an egg in a bed. Nothing not for.
Depends on how well you want it done.
Okay.
I'm a soft boiled.
Should we take it out?
Now it's like how long in? 1.5 minutes probably. So we wait 1 more minute. No, like in total like 3 minutes.
Yeah, you don't want to run it. No.
So 21.
You want it hard boiled, right? Or
So inside this mesh scoop is an egg that we're cooking for
Anywhere there is not gonna take
school.
much longer to
A school experiment.
Should we try to measure temperature? To measure temperature at that location? Or is it not really?
try it. It's gonna be real.
Okay, we can
It's
we can take out the egg now. Yeah.
How long has it been there?
3 minutes probably.
3 minutes?
Yeah.
Okay, we'll see. Cool. It's
It's
still smoldering.
Still.
That's excellent. Good.
Excellent. Yeah, take your thing.
How difficult would it be to measure temperature?
Yeah, we're going to try and get it to go.
Yeah?
It's
Okay.
very close, though, so it's...
We just don't want to, extend too much beyond the time, of course, where we're supposed to come up. So I think we have like 8 minutes left.
But you would need it for the rock cleaning.
So watch that on YouTube, right? Yeah.
I have it. I can't see the
Well,
temperature sensor, let's see.
It's right there.
That helps.
Right there.
Oh, yeah. It could be, but it's still cool.
Right. We're going to clarity. To your left. You got it.
But I can't get it. I can see it.
Pretty sure. Go straight down.
I think you got it now, yes.
I get it.
Yeah.
Nice.
Ah. You can off the wall, Chris.
Alright.
All right.
See if we can pick up.
Another can. I don't know. It's catching on the something
Cooler.
cooler, probably.
Did everything put that in, just like
Yeah.
drag it back?
Yeah, maybe. Yeah. Oh no.
You try and
What's that?
try again.
Nope, it's catching.
Try again.
No, hold on. You just put it on. Yeah, it's not bad. It's where it is.
Okay, I'm sorry, we can't get temperature from, I can't see it.
Yeah, no, that's fine. Yeah, I mean that's the way it is.
Okay.
Hopefully, it's not going to be a problem that it's hanging low.
That'll be fine.
Okay. All right.
Sorry, 2 seconds, Chris. You need to go on the heavy somewhere.
You need what? It's fine. Angle.
The other one's down as well.
Yeah, we can. It's what it is. I can't torch in, so I don't know what we'll do about it right now.
What's the channel?
Shouldn't have.
Thanks a lot. It was challenging.
I don't know what happened there at the end with the box cuts.
Can you give a quick dive sign off? Can you give a dive sign off?
Oh. Yeah, so this was the... Yeah, this is Stefan Siebert again from Microbial Oceanographic Institution, reporting here from Falco 2. This was our dive today with Sebastian in the water. Was a successful dive overall with some challenges in between. But we started out at Teddy Bear up north, recovered an experiment, took some samples along the way at a diffuse flow site called Fuzzy Rock. Did some work at our experimental site, checking on the mesh boxes that are designed to test the hypothesis that Riftia or other organisms might actually recolonize from the subsurface. And then we proceeded to obtain fluid from Grubbs Bar, subsurface fluids. Also collected some Riftia and finally ended here at the Black Smoker at Bio9 and P-Vent, two really massive structures that are really impressive, venting hot temperature fluids. And so we were trying to get a couple of samples. We were successful in getting a suction sampler from an Alvinella colony and maybe some pieces of a rock. And in the end, successfully boiled an egg, hopefully, as well. So thanks for joining today. It was a pleasure to have you come along, and hope to see you back or hear you back or tomorrow. Goodbye.
Yeah.
Yeah, we got your meeting.
Thank you guys. Again, if you are streaming on Facebook, we will be ending the stream because we have reached our 8-hour limit. If you would like to see the ascent, you can move over to Schmidt Ocean Institute YouTube, and we will be streaming the ascent there. Thanks again. We'll be diving at the same time tomorrow, and we'll see you then.
Yeah, we're gonna do it. If we need to do something, we've got extra hinges and stuff, so we can rebuild it. Yeah, no, I understand.
Okay, for those of you on YouTube, we are just going to mute the control room as we make our ascent, but we will again always focus on any critters we see on the way up. Thanks for sticking with us.
Thank you for joining our midwater ascent streaming here in the Underworld of Hydrothermal Vents expedition. We're reaching the conclusion of our dive, about to do our recovery, less than 100 meters of seawater between us and the surface. We'll keep the cameras going. We might see another oceanic white-tip shark. But thanks for joining us. We'll see you tomorrow. Plan to launch the same time, 6:30 ship time, Costa Rican time. And we'll see you then.